question, therefore, is do the ECHO guidelines, which have only changed marginally from 2015 to 2022.
طبعا فالدكاترة بيشوفون مرضى ضغط الشريان الرئوي بأنواع مختلفة.
وكانت في البدايات يعني ما في تحديد لأنه أنتي فاسفولبيت كانوا معتبرينها تخثر دم عادي. واستمريت على الورفرين.
Regulating night, for instance, BMPR2 loss of function,
genetic drivers, mediators like growth factors.
Have a chance of individualized therapy, measure their risk based on implantable monitors.
Stimulates endothelial angiogenesis and formations with calyx for expression compared with control.
System of the organ check. It's a very small one, so I'm wondering the number.
Study links to Wilson in heart failure patients. To matrix. But when you put an ETO ownership with the size, so
give a couple of more minutes. Okay.
Have a role in the future in that it's in a very, in an expert center doing.
And what do you think the inhaled imatinib
updated guidelines from the definition to the risk stratification to the impact on the treatment.
It was used in different ways by different centers and by looking at feasible treatment given for these patients. So the question is that relevant to the patients?
Often the patients who started that study being on the ERA PDE5 inhibitor combination background.
Increasing pressure, recapillary, increasing resistance.
Everybody, and you have patients who are even functional class 2 doing fairly well, and you have patients who are
is really important, and I see that it really
being a cardiologist and a heart specialist.
The cause, initial cause, and it's insisting on a very unique population of elderly males.
6-minute walk test that you're looking at market worsening lesions, but vasculopathy is there.
Basically phenotypes. So again, that is supered from the data from both Compare and the Aspire.
Utilize according to different treatment strategies.
But these are the metrics that you have potentially changing the recommendation level.
In Victoria was very peculiar in the way that that's the overarching mechanism. Then some patients derail, and so the chances
of them developing osteoporosis is, I mean, in the way that we're talking about a disease that it has a lot of high PVO.
When Compare data was published, I think that was notable, get you that far. I think in the future, looking at parenchyma.
Going to be very exciting live cases.
Thank you.
Difficult decision to make a decision to send the patient for right heart cath. So
He was 813. His 6-minute walk test, he had a saturation of 94%. The disease associated PAH can be also improved in addition to the vasodilatation CTD in PAH.
Thank you for showing this case.
This may induce quite rapid.
Class 4 upon her presentation. Her symptoms was associated with orthopnea.
Also, if you go look for another article on people who had people.
One of the key approaches for such patient, you try to optimize their situation.
Diagnosis, then the second part, inshallah, will be a follow-up for the case. Her CT showed bilateral, non-specific ground glass.
Organizers for the fascinating meeting as usual, and the very nice program and the entertainment. Everything actually is.
Basically, utilizing a large healthcare data, insurance, clinics, hospital episodes, emergency visits.
I think it's about time to start. Well, Assalamu alaikum everybody and good morning. Bismillah, alhamdulillah, wa salatu wa salam ala rasulillah. So I think we have a decent number of attendees, and thank you all for coming. This session today is about remodeling and updates about important studies that have been making, I would say, a breakthrough the last couple of years or 3 years. It will be covered by outstanding international speakers. So the first, of course, I have to tell you that Dr. Anna Jimenez lost her mom recently, so she had to stay with her family. So I'm covering, I'm cheering for her. Dr. Jing, I was called by Beijing yesterday, so he had to leave. So Dr. Mark Humbert is covering for him. So it is my pleasure to, can you turn off the music, please? Yes, it's my pleasure to co-chair this session with Professor Mark Humbert. And I will start introducing him. I don't know if he needs an introduction. So Professor Mark Humbert is one of the pillars of the pulmonary hypertension worldwide. He is from Paris, and he is going to talk about reversing and modeling a potentially achievable goal. Mark.
Thank you. Thank you very much. It's very loud. Thanks very much for your kind invitation. Always a pleasure to meet you and your group and all the friends in the room and online. It's a privilege to be invited to give an update in the remodeling reverse hypothesis and to try to show you that in the last 30 years we have built a lot of information which demonstrate that it might be indeed a possible achievement to reverse pulmonary vascular remodeling. So thanks to SAPH for the organization of this global meeting. So I have some disclosures. And I would like to start with some comments about the process of World Symposium and when did pathology and pathobiology really took the light in the last 60 years. So I think other speakers have already presented similar slides, but as you can see here at the bottom, the very first time we had a dedicated chapter on pathology and cellular and molecular pathobiology was in 2003 in Venice at the World Symposium. And you can see among the names some very distinguished colleagues were all very involved in the understanding of pulmonary vascular biology, and the concept of pulmonary vascular remodeling was already very high. Since 2003, every 5 years, we have a dedicated task force on pulmonary vascular pathobiology. What I would like to show you, it's the very first chapter we published as a group on the pathobiology of pulmonary hypertension. And I just wanted to give you an overview. I'm not going to go into the details, but as early as 2003, the whole scene was set. The whole scene was set. You see here, bottom left, the 3 pathways, which were very much discussed in the next 20 years with drug development. Here in the middle, the serotonin pathway, which is so complex and which led to many, many negative trials. That being said, serotonin remains an important pathway in PAH. On the right-hand side, already in 2003, the BMPR2 knowledge was quite advanced. And here on the top right, there was a summary slide showing the complex interplay between gene, environment, and cellular and molecular disturbances. I just put one more slide in the spotlight because in the first article in 2003, published in 2004, you see that there was already a figure on the potential role of the TGF-beta superfamily in pulmonary vascular remodeling, with the keywords TGF-beta, activins, BMPs, and GDF. So it's very reassuring that we know where we come from and that it was already in the spotlight more than 20 years ago. So year after year, we meet at World Symposium, we meet at great meetings like today, we meet at American, European, international meetings in Japan, China, etc. And you see that the World Symposium is always a kind of peaceful time where we can meet in a relaxed way. And during these very open discussion, and there are too many people missing on the slide, and my wish would be to enlarge the groups, but you see on this slide, there were many people involved in pathology, pathobiology, and we were asked to summarize every 5 years what has been achieved and what are the research perspectives. And in 2019, after the World Symposium in 2018, this summary slide was published, and you see that many keywords remain as compared to 2004 with the environment. You see here mechanical strain, hypoxia, nutrients, the background genetics, and the complex interplay between growth factors, transcription factors, metabolic state, and epigenetic modulators, leading to a pulmonary vascular remodeling, which is only one way on that slide, but the objective is to reverse remodeling, and we need to understand how it happens in order to reverse it. You see, same picture, same order of people. We set the scene, and it was last year in 2024 in Barcelona. You see Marlene, you see Sonny, you see many people on the Christophe here, Peter. You see many people who get older, but who are getting more mature and with more and more insights into the pathology and pathobiology. And I borrowed this slide from Martin Wilkins' summary slide, which indicates what are the main drivers of PH pathology. And it's a very simple cartoon. On the left-hand side, you have the normal vascular lumen, which is very soft, a very thin wall with a monolayer of endothelial cell, not much smooth muscle cell, and a soft adventitia. And you see on the right-hand side what's happening in PH. And in PH, you have many problems. But we need to find bottlenecks. And that's a key word, bottlenecks. You need to find sites where you can intervene and in an attempt to block progression and, if possible, reverse progression. And I'm not going to go through all the slides because we don't have enough time, but I would like to say that always, and I see lots of people in the room involved in genetics, always genetics indicates a key pathway. It doesn't mean that it explains everything, but when you have a condition caused by a gene mutation, it is very likely that this gene mutation has an impact on the pathobiology. And similarly, when you have many genes, you have many ways to understand the pathway. And our enemy is pulmonary vascular remodeling, this very remodeled vessel. And we need to find the bottlenecks. And in 2024, I would say that the most important bottleneck we have identified is the TGF-beta superfamily. But there are others, and you will hear more of PDGF and other pathways later today. But TGF-beta is very important because it is a key factor of vascular homeostasis. And you need to have a very well-balanced signaling between the SMAD2/3 pathway and the SMAD158 pathway in order to have a fine-tuned pulmonary vessel which will not progress to remodeling. And in PH, we know for sure that the TGF-beta related mutation BMPR2 alters the TGF-beta balance in a direction which favors pulmonary vascular remodeling. However, carrying a mutation is not sufficient. You need other factors, and that's a complexity of science. Here I have super simplified the cartoon. You see here the pulmonary vascular homeostasis with a fine-tuned SMAD regulation. And here you have the upstream drivers. You see on the left the BMPR2 SMAD158 pathway, which is anti-proliferative. And here on the right, the activin receptor type 2 SMAD2/3 pathway, which is pro-proliferative. And we know for sure that in PH, there is that mutation which triggers the vast majority of cases. And when you have this mutation, you will certainly favor the imbalance between TGF-beta pro and anti-proliferative pathway. So at the World Symposium, with Christophe Guignabert leading the group, we put together some slides and some concepts which were extremely important for educational purposes. And you see here the key word on this slide is that the pulmonary vascular remodeling is driven by three interconnected forces. On the left, you have the endothelial cell dysfunction, which is prominent in PAH and which has been the reason for understanding the prostacyclin pathway, the endothelin pathway, the nitric oxide pathway, and more recently the TGF-beta related pathway. In the middle, you have fibroblast and smooth muscle cell dysfunctions, and on the right, inflammation and disimmunity. These three interconnected pathways are certainly disturbed because of genetic and environmental factors, many related to TGF-beta and exposures. On this next slide, you see that the dysfunctional pulmonary vascular cells are characterized by an array of abnormal signaling, but among the array of problems, you have hits, and BMP TGF-beta is important with many others, and the cellular consequences, which are markers, key points of pulmonary vascular remodeling. And among the mediators which are more and more demonstrated as involved in pulmonary vascular remodeling, you have the growth factors led by TGF-beta, PDGF, FGF, etc. Cytokines led by pro-inflammatory cytokines, but also other cytokines like activins, and BMPs and activins. And the pulmonary vascular remodeling is a consequence of this very complex mechanism. And once again, we need to identify what can be targeted safely. And this is the message we need to have. So right after me, we will have a great scientist, Paul Yu from Harvard Medical School, and he will spend some time on the TGF-beta pathway. I'm not going to go too much into the detail because you will hear more about that. But once again, my initial cartoon was an oversimplification, and this cartoon is also an oversimplification. So it shows you that everything is complex, and when you develop a drug interfering with these pathways, you need to be lucky. And we're discussing that over breakfast with Paul Yu. You need to be lucky, as always, but also you need to be smart and identify the problems when you interfere with the pathway with some good and bad effects. So what is our goal? Our goal is to maintain pulmonary vascular integrity. The reason why our patients are short of breath is because their small pulmonary vessels are occluded. Sometimes because of constriction, most often because of vessel limitation and obstruction due to remodeling. And you see that the SMAD158 positive and the SMAD23 negative drivers need to be targeted in a safe way, and we know for sure that safety will be a key word in the development of TGF-beta related drugs. So in PH, we know, and it's now very well established, that there is a disconnection between SMAD158 and SMAD23, and that there is a clear overexpression of SMAD23. So the decrease in SMAD158 related to BMPR2 and BMPs is only a small part of the story. The other part of the story is overactivation of the activin pathway, and we have data in humans. We published with Christophe 2 years ago this paper from humans, and we showed very clearly that in humans, irrespective of the genetic background, even in non-mutation carriers, there is an overexpression of inhibin beta A, inhibin beta B, meaning that activin is overregulated in the small pulmonary vessels, leading to remodeling, or at least contributing to remodeling. Then our colleagues in Japan, I know there are some Japanese friends in the room, our colleagues in Japan from Kobe developed that very important study published in Nature Communication, where they showed clearly that when you delete activin or when you overexpress activin, you induced either a limitation of remodeling or an acceleration of remodeling. So it's very complex, but if you look for a bottleneck, activin may be a bottleneck. And of course, there are many, many ways to interfere with the pathway. And the pathway is so complex that you need to be clear in your mind when you develop a new drug. So I'm not going to discuss all the drugs, it will be done after me. But clearly, if you want to stimulate the BMP branch, you can use recombinant proteins, activators, and gene therapy. Some are in a mature way ready for drug development, but you can also block the pro-proliferative pathway with monoclonal antibodies, ligand traps, inhibitors, and antisense. And you know for sure that the ligand traps has been developed with good success. In PAH and in experimental model of PAH, we know that Paul Yu's lab showed that activin receptor 2A FC, so a me-too of sotatercept for animal models, can limit progression and even reverse experimental models of severe PH. And we know in humans, and we have published this fall, this proteomic study, when you treat a patient with sotatercept, you induce not only changes in activin, but also changes in more than 50 proteins which are involved in cardiovascular, metabolism, cell regulation, inflammation, and immunity. And that's very important to understand how it interferes with the pathway. So before closing, at the end of the morning, you will have a lecture on targeted therapies. And I wanted to emphasize that many growth factors are involved in the progression of pulmonary vascular remodeling. And PDGF is really a treatable pathway which signals through two main pathways and which induces the proliferation and migration of smooth muscle cells and fibroblasts. And there is for sure, and you see here that figure from Frédéric Perrault in my lab, there is clearly an overexpression of PDGF, PDGF-beta, PDGF-B, PDGF-A, PDGF receptors in the small pulmonary vessels of PH. And we know that we can interfere with the PDGF pathway with tyrosine kinase inhibitors. So you will hear more after my talk. So in summary, it's complex, but it is understandable, and you need to be always open to surprise. And in the last few years, some obvious concepts were contradicted by the facts. So you need always to be ready to understand what's going wrong after experiments, not only in preclinical models, but also based on clinical trials. Among all the pathways, the BMP-TGF-beta pathway is very prominent right now, and there are drug developments ongoing, but other pathways are important. So right now, the three main pathways we usually take care of for the treatment of our patients remain the treatment of choice as first-line therapy, but then as add-on therapy, you have these emerging solutions which appear very promising, but we need more research to better understand and refine the bottlenecks we can interfere with. So thank you very much for your attention.
Thank you, Mark. As usual, outstanding talk. It's my pleasure now to introduce our next speaker, Dr. Paul Yu from Harvard Medical School. He's going to talk about TGF-beta, BMP, and other interacting signaling pathways and potential targets for new therapies. Paul?
Thanks so much for the opportunity to speak here and to join this wonderful symposium. I just wanted to go over our conflicts and start pretty much where Mark left off. Thank you, Mark, for the really great introduction to the background of how there's such interest in this pathway as a target for pulmonary vascular disease. And no doubt we owe a lot of this progress to the wonderful genetics work being done in this field over the past 2.5 decades by people in this room. And of course, we all know that of the many genetic loss of function mutations that are found in heritable pulmonary hypertension, as Stefan Graf really beautifully summarized yesterday, there are many other mutations outside the signaling pathway involving ion channel transport, water transport, developmental patterning, and other functions that may be coordinated with BMP and TGF-beta signaling, may have really important crosstalks with BMP and TGF-beta signaling. But with all the BMP TGF-beta mutations, you notice that they are all concentrated on the BMP signaling side, this right side here, which use the sort of classic BMP receptor, the BMP type 2 receptor, in conjunction with various type 1 receptors. And the mutations all focus on a signaling complex that's specified by BMPR2, ALK1, the co-receptor amyglin, and a variety of ligands that have high affinity for these receptors, notably BMP9, BMP10, which turn out to be loss of function loci as well. So if we had had the benefit of this set of genetic clues, I think that progress might have happened a little bit faster in this field, but nonetheless, I think this puts us in a really nice position to start understanding how we might be able to modify the pathway, although, like Dr. Humbert said, this is a complicated pathway and there are lots of surprises as you start tinkering with it. And one important message to take home from this is that while BMPs are very important for pulmonary hypertension, the pathway as a whole is very important for vascular disease and vascular development in general. On that same right side of the pathway with the BMP signaling effectors and receptors, not only do you have pulmonary hypertension, but you also have heritable hereditary hemorrhagic telangiectasia, as was discussed yesterday in our sessions, overlapping with several of the same loci. So these are overlap syndromes, and they have very different pathophysiology, very different complications. HHT is 100% penetrant, and we know heritable PH is only 20 to 25% penetrant. But look at the overlap in the loci. We also have, in addition to these genetic loss of function hits, we have acquired loss of BMP9 and 10 through hepatopulmonary syndrome and portal pulmonary hypertension, and then loss of GCN2 or even BMPR2 can cause pulmonary veno-occlusive disease. And then on the left side, you have loss of function with some of the TGF-beta activin GDF signaling machinery, and these can manifest in some of the syndromic aortopathies like Marfan's, and Loeys-Dietz syndrome can manifest as congenital heart disease in many forms. And also, there's some evidence now to suggest that some of these loci may be protective, certain polymorphisms may be protective in atherosclerotic cardiovascular disease. So I think it's important to understand the greater context and how this pathway really is so fundamental to cardiovascular health and development. So I'm not going to talk a lot about the story behind sotatercept today, because we've spoken about it a lot in the past in this forum too. But this was a really remarkably fast translational story due to the fact that this was a repositioned asset. Sotatercept, or Actor 2A FC, a fusion protein, blocks GDF8, GDF11, activin A, and activin B. And it was developed for anemia and also explored in a lot of solid tumor oncology trials. And we knew that it was a very potent drug for raising hemoglobin, but it was a bold move by the company that had developed this molecule, Acceleron, to try and pivot to pulmonary hypertension. And when we started looking at this in preclinical models, what we found was that, as Mark showed, the effects were quite potent in preclinical models. It seemed to not only prevent the obstructive remodeling and obliteration that you saw in some of these models, but it also seemed to recanalize vessels that had already been obstructed. And then that led to a very almost contemporaneous phase 2 trial, Pulsar, which found quite remarkable efficacy. And so what were the mechanisms? I think that there are effects on all the cell types that Dr. Humbert just reviewed. There are effects on endothelium, there are effects on fibroblasts, adventitia, as well as smooth muscle. Histologically, we appreciate most a decrease in inflammation and a decrease in medial hypertrophy with the treatment. And this is even in a very, when we set the bar very high with the delayed treatment, where we're allowing pressures to get almost to systemic levels in the rodents. And we try to start treatment when 10% of the vessels are already obliterated. And you can see that at the end of 9 weeks, some of the vessels are seen to be patent again. This is associated with decreased proliferative index in the vessel wall and a resensitization to apoptosis in the vessel wall. There seem to be apoptosis-resistant cells in these pulmonary hypertension models, and that seems to be restored to normal. But I won't go through all the data, but I think this is another important note. Activin is a fibrotic molecule. It has been appreciated in the renal literature for quite some time that it promotes fibrosis in many tissues, but it's not something that you can equate to the effect of TGF-beta. Even though they share some of the downstream effectors in the cell, somehow the cell is able to read and interpret the signal as being something quite different than TGF-beta. You have activation of SMAD2 and SMAD3 in cells like fibroblasts or smooth muscle cells that are stimulated, and you certainly get the expression of a lot of myogenic and fibrogenic genes in your smooth muscle cells, but the profile of those molecules that is induced by activins, GDFs, is quite different in terms of the proportion of these molecules than of TGF-beta. So even though these functions are specified by a very small number of effector molecules, SMADS2, SMADS3, SMADS4, there is a lot of subtlety going on at the intracellular level to interpret the functional output. So these are unique molecules in terms of promoting fibrosis in the extracellular matrix remodeling. So I told you that sotatercept was developed as an anemia drug, and it was noted in the phase 2 trial already that there were some limitations in the populations we could dose due to the rise in hemoglobin. The regulatory authorities had set the maximum dose at 0.7 milligrams per kilo for the fact that that would be predicted to raise hemoglobin 2 points over a month or two. And if you look at the distribution of hemoglobin levels in our group 1 PAH population, you see that the lower quartile are actually anemic and would stand to benefit from these hemoglobin raising effects, but the upper quintile would be excluded from taking sotatercept right from the outset. And arguably, even some of the patients who are in the 14-16 range could easily get into the danger zone in terms of polycythemia. So this is one limitation, but then it became clear also that telangiectasias were happening during the open label dosing of this. And this became more evident in the phase 3 trial, where not only were telangiectasias seen, but with heightened scrutiny, attention, epistaxis, telangiectasias, and even GI bleeding episodes were observed. So one question is, could this have been anticipated? Because this was not really a common feature in any of the 400 patients that were dosed with sotatercept prior to being used in pulmonary hypertension. What do we know about the experience? So, and the biochemical profiles. Acceleron had 2 other molecules that they had looked for in various applications for anemia. And sotatercept was actually the first molecule that they looked at. And then they looked at ActR2B, and then they looked at a modified ActR2B receptor that had been edited slightly to eliminate BMP9 and other ligands. And there was actually no bleeding in luspatercept, which was then approved for anemia, for MDS, myelofibrosis. But ActR2B actually caused nosebleeds in subjects within a couple of weeks, and also even in some healthy volunteers. And ActR2B is a receptor that is a little less abundantly expressed than ActR2A, but it binds BMP9 and 10 very avidly. And so it turns out that that might be the explanation for some of these observations. In fact, there were molecules generated by Pfizer and also by Acceleron and TRACON that also had potent BMP9, BMP10 inhibition activity, and those were all associated with epistaxis and telangiectasia. And if you go back and you look carefully at those trials, some of them were also associated with pericardial effusion. So I think the consequences of having some residual activity for BMP9 were that in relatively healthy populations, sotatercept looked to be quite safe. But what we are learning with prolonged experience is that there might be some catch-up with chronic dosing. Could we have detected this in any of our preclinical models? It was pretty subtle, but when we look back at some of our studies, what we noticed was that some of the rats that were not healthy rats, but rats that had received Sugen hypoxia, so probably have some degree of systemic vasculopathy, developed hind limb swelling. And sometimes unilateral, sometimes it was bilateral, and hard to pick up except from a picture when it's quite obvious, easier to measure when you do some water displacement plethysmography. And you can see that there's generally a trend and sometimes a significant increase in hind limb to femur length volume. But I think that there are more sensitive assays. A lot of the HHT models were developed about 20 years ago by Helen Arthur, Michel Letarte, in a strain that was used to generate the original genetically modified mice. This is SV129. This is because they were the stem cells that were available for making transgenic animals. And it turns out that this strain was a fortuitous choice because it's very prone to manifesting HHT. And if we administer sort of a modern day derivative of SV129, because the original strains actually don't exist anymore, not commercially, we find that potent BMP9/10 traps like ALK1 and Actor2B, both which had not passed safety in studies for DMD and for cancer biology, actually cause varying degrees of AVMs, including mild telangiectasias in their paws, and even some bleeding complications. And this can be so severe that the animals will develop anemia. And you can see here that if you were paying close attention and you had run these trials in sufficient power, that sotatercept, or its analog, would have given you something significant over isotype control, and then anti-BMP and anti-BMP9 and 10 by themselves would give you more subtle effects. So we think that this drug-induced HHT model might do something to help you understand the risks of some of these HHT mimicking effects. Interestingly, the tail also has differential susceptibility to these different ligand traps and neutralizing antibodies. And when it's severe, there's bleeding or even just tail necrosis. I don't know what this means for people. We don't have tails, but there are probably tissues and specific species-specific differences that make the consequences of these different traps different from human and mouse. So where does that leave us? Well, I think, sotatercept is really still such a powerful clinical tool, and I think that we're going to learn ways of helping to minimize the toxicity because it doesn't happen to everybody, and there might be some clever dosing regimens that would allow you to sidestep this while still preserving some of the gains that you've had while being dosed. But we wondered if there are other ways of targeting this pathway. And this paper from the Humbert group led by Christophe Ginebert and Sabine Bailey showed really nicely that BMP9, surprisingly, is a potential target for therapy. So they showed in this paper that ALK1 FC, which I told you before didn't work in clinical trials due to bleeding, was a very effective way in treating existing pulmonary hypertension due to monocrotaline or Sugen hypoxia, always given after the establishment of disease and nicely improving pulmonary hypertension and RV hypertrophy. And consistent with this, BMP-deficient mice were resistant to hypoxic pulmonary hypertension, and delivery of an anti-BMP9 molecule was somewhat protective too. So we wondered, is this an effect that is dependent on the pathology of existing pulmonary hypertension? Could what we assumed was a protective molecule, turn into a pathogenetic molecule in the disease context? So this was the first experiment we did. We used the same ALK1FC, and we gave it before challenge with Sugen hypoxia, and then we gave it after Sugen hypoxia in the way that, that Christophe and Sabine did. We were hoping that by giving it before vascular injury, it might exacerbate pulmonary hypertension. But what we found was that these two doses given even before the onset of injury were actually mildly protective. In contrast to the 6 doses given after injury, well, it wasn't as good, but at least there was some durable effect enough to show that it might trend towards protection. So it seems like whatever context you block BMP9, at least in rodents, blocking BMP9 and 10 is protective. Well, then we thought, well, maybe we're missing something here. We know that Sugen, an anti-angiogenic molecule combined with hypoxia, exacerbates pulmonary hypertension. What would happen if we gave ALK1FC with hypoxia instead of Sugen? Would it mimic the effects of Sugen? So what happens if we give it concurrently with this model? And it turns out, just as Christophe and Sabine showed in the mice, that in the rats, hypoxia and ALK1FC do not synergize like Sugen. In fact, it pretty much normalizes hypoxic pulmonary hypertension. So you can give it before, during, or after experimental disease, and it seems to be helpful. Now, we know that ALK1FC is not a strategy, but what about anti-BMP9? Because Sabine and Christophe showed that it might be helpful. And would it give you efficacy that would be noteworthy enough to want to continue and maybe move towards a human trial? So we compared the Actor-2A-Fc or sotatercept equivalent molecule side by side with ALK1FC and this commercial anti-BMP9 antibody called MAB3209. And we looked at a 4-week endpoint and a 6-week endpoint. And what we found was that there were protective effects that were pretty equivalent from all 3 of these strategies. Very surprising, because you have sotatercept, a molecule that blocks maybe 14 or 15 of the ligands of this pathway. That's half of the 33 ligands. And then you have a reagent that blocks 9 and 10, which we already know is a sort of danger zone for pharmacology of this pathway. And then you have BMP9 neutralizing antibody, which just blocks a single ligand. So how can that be? Well, it bears out even with longer-term treatment, and with remodeling that the anti-BMP9 seems to be equivalent to the broader, less specific strategies. So is there some basis for expecting this? This nice work from Mark Ormiston's group in Queen's College showed that maybe there is a context-dependent effect of BMP9. They found that BMPR2 wild-type endothelial cells had a nice anti-proliferative response to BMP9, that heterozygous mutant cells had a loss of that anti-proliferative response, and that homozygous knockout animals had a pro-proliferative response. So it's possible, at least in the context of pathogenetic mutations, that the normal cell cycle control that might be provided by a protective, supposedly protective molecule like BMP9 might be lost. In fact, we see this difference and show this in the sotatercept paper, that cells cultured from diseased lungs are actually prone to gaining this pro-proliferative effect in response to BMP9. But what is downstream of BMP9 in endothelial cells? So it turns out that a lot of the molecules that we associate with pulmonary hypertension pathobiology are direct transcriptional targets of BMP9 in pulmonary microvascular endothelial cells. And many of these are shared with the transcriptome of diseased human lungs with pulmonary hypertension at the time of explant. These include VEGF-A, endothelin-1, IGF-BP4, and PDGF-A, and to some extent PDGF-BB as well. And you can see in this volcano plot that these are the upregulated genes. We know that endothelin is a validated target, and actually Christophe and Sabine showed very nicely that endothelin may be responsible for part of the effect of protection by BMP9 neutralization. We went a little bit further, and we integrated the results from a couple of large single-cell and bulk RNA-seq data sets. And what we find very consistently across these data sets is that CXCL12 is one of the more significantly upregulated genes downstream of BMP9. in cultured cells, also in diseased human lungs, and diseased human lungs by microarray, but specifically in the endothelial cell clusters in single cell. And then it's also one of the most downregulated genes when animals are treated with anti-BMP9. So it certainly seems that CXCL12 might be a potential therapeutic target. And it's one of the 3 genes that actually is found from the intersection of these 4 data sets. So CXCL12 is highly upregulated in a lot of different cells from PAH lungs, but it seems to be more highly upregulated in the endothelial cluster. And if you break down the endothelial cluster in these single-cell data sets, this is from the Lafayette's group, it's upregulated in GCAPs, in arterial ECs, and systemic venous ECs. And it turns out that you can culture any pulmonary microvascular generic endothelial cell and get potent induction of CXCL12 expression. And it turns out that CXCL12 is a powerful fibrogenic and mitogenic stimulus for smooth muscle cells. You can see the phenotype shift that we associate with the pathologic signature is potently induced by CXCL12 in cultured smooth muscle cells. And we could postulate that this might be a process that's initiated by these diseased endothelial cells in disease. And this is actually the co-culture result that Ying Zhang did to try and prove that, where she found that smooth muscle cells in monoculture did not respond to BMP9, as you might expect, and had no CXCL12 expression, no phenotype modulation. But when co-cultured with endothelium, BMP9 caused a phenotype switch to express calponin. This could be neutralized by a neutralizing antibody to CXCL12 or by an inhibitor to CXCR4, the CXCL12 receptor. And the effective human serum, which similarly induces calponin expression in smooth muscle cells, could also be blocked by anti-BMP9. I don't have time to show you all the potential pathways that BMP9 appears to be impacting, but they include at least a subset of these candidates. There is a clinical stage compound that blocks BMP9 that's being advanced by Pfizer. We think right now that BMP9 is sort of a Janus-like molecule that has obviously important roles in cardiovascular and pulmonary vascular development, but in the context of pulmonary hypertension takes on this disease progressing role, and that the downstream effectors of this effect probably involve some of those BMP and TGF-beta effectors that probably gives its dual identity. And we think that the SMAD2/3 effects might be particularly important in disease. So I can tell you that this asset from Pfizer has now advanced to phase 2, having already gone through phase 1 safety studies. I think that this proposition gets people nervous. I think the anxiety is probably well placed, but I don't think that we can make progress in this field if people don't continue to make bold moves like this. Why do I think this might work? All the agents that have been now associated with HHT, like mimicry, have potent inhibition of BMP10 as well as BMP9. Our studies show that in isolation, either of these ligands might be acceptable for inhibition. So the hypothesis is that this anti-BMP9 molecule might be able to thread the needle just enough to get you some safety, but still very potent efficacy. So that's the hypothesis. I'll summarize by saying we can model HHT-like effects in susceptible strains. It seems that blocking 9 and 10 are the most important factors. We think that BMP9 exerts a lot of paracrine signaling in the vasculature to promote pulmonary vascular remodeling. It turns out to be every bit as difficult to tease apart just as the activins, but we're only focused on one ligand, which makes it a little bit simpler. And we are eagerly awaiting the next steps in sort of advancing this to the clinic. I just want to conclude by thanking all the people who contributed, including Ying Zhang and Peiran Yang. Peiran is leader of a group in Beijing now. We had a lot of help from the Pfizer team and many collaborators. Thanks so much for your attention.
Thank you very much, Paul. Please join us to was a great trip together through the TGF-beta pathway. So now we move on and we remain in Boston with Professor Johanna Preston. Johanna is going to discuss sotatercept in PAH therapy, update on long-term data and possible role in other forms of PH. So the floor is yours. Thanks.
Thank you. Good morning in the last day of the wonderful symposium. Thank you so much for the organizers for inviting me again. It was a pleasure to come back. And continuing and translating the TGF-beta superfamily BMP story from very complex and interesting preclinical work and moving on to patient science, it reminds me that this painting reminds me of what was the type of care we used to provide to our patients with pulmonary hypertension when I was a student. And it was just compassionate, holding hands, and soft talking. And then now, as Sean Gain showed yesterday, the treatment algorithm that is recommended for patients with pulmonary hypertension includes combination therapy, but also the additional fourth pathway that can be tackled in pulmonary vascular disease with the activin signaling inhibitor, for which we have a first therapy with sotatercept. And if you look at the recommendation is to consider one of the options to add an activin signaling inhibitor in patients who are on background therapy but not at goal at the time of assessment, whether they're intermediate low, intermediate high, or high risk. The fourth pathway can be added. And this, as Paul showed, and also Mark alluded to, was based on the pivotal clinical trial STELLAR that showed efficacy of sotatercept in the double-blind randomized control trial with sotatercept versus placebo in 323 patients. But when we add a new medication and a new pathway and a new target in a rare disease, especially in a rare disease, we like and we want and we need to accumulate long-term safety data to better understand what are the patients that mostly benefit from this additional therapy and which are the patients that we should avoid introducing therapy because they're at high risk of adverse events. And this brings me to the Soteria long-term open label trial that was built to accumulate all the patients that were initially concluded, the several clinical trials that tested the efficacy and safety of sotatercept. Pulsar trial was the phase 2 randomized trial. STELLAR was the pivotal phase 3. SPECTRA was a small open label trial geared at assessing exercise capacity with a cardiopulmonary exercise test. Hyperion, earlier disease with earlier diagnosis, and Zenith, more advanced disease patients enrolled in this double-blind clinical trial. The majority of patients that at the time of the interim analysis of Soteria included patients from Pulsar, STELLAR, and SPECTRA. And now that the Hyperion and Zenith are recently closed, we will be offered enrollment into the Soteria clinical trial. Talking about other groups of pulmonary hypertension that may benefit from modulating the TGF-beta and activin pathway inhibition, Cadence is a randomized clinical trial that is testing sotatercept in patients with pulmonary hypertension associated with heart failure with preserved EF. So this is a group 2 clinical PH clinical trial that is ongoing. So this would be another type of vascular disease that we will have more information. Lightray has a fixed dose of sotatercept, and also there's an open label phase 2 pediatric PAH that is testing the activin signaling inhibitor. So this is the Soteria study design, where patients are enrolled from the parent study and started on long-term open label. Sotatercept, the primary objective is to evaluate long-term safety and tolerability, but there are secondary efficacy side effects that are being measured. And this is the first interim analysis that will be recently in publication in European Respiratory Journal. The study is supported by Merck, and what I am going to present are the results on behalf of the co-authors. These are the baseline demographics of the first 426 patients, the average 49 years of age, majority female patients, and with idiopathic PAH, with functional class 2 and 3 in the majority of them, and heavily treated with background therapy, most of them on double and triple therapy, some with prostacyclin infusions. This is the exposure time to sotatercept with a median range of over 2 years, ranging from 3 weeks to 5.4 years, and over 900 patient years exposure. So focusing on safety and tolerability, if we look at treatment emerging adverse events that led to study discontinuation or death, to study discontinuation, 16 patients, and then there were 12 patients that died, and I will go over more. None of the events were attributed to the study drug. Now, if we look at the 13 patients with serious adverse events leading to study discontinuation, there were 13, and then those leading to death were 12. The paper will go into more detail over all these patients, their comorbidities, and background therapy. Knowing, as Paul said, that there is an increased risk of bleeding, if we focus on the 2 patients with gastrointestinal hemorrhage, these were not events that were associated with the study drug, nor the other 10 patients' events. So let's focus on bleeding events. The bleeding events were indeed the most frequent adverse events of interest. They occurred in slightly over 1/3 of patients. The majority were epistaxis events. If we look at the 22 serious bleeding events that occurred, there were 4 patients that had bleeding events related to treatment, and 2 of these patients required discontinuation of the treatment. Moving on to thrombocytopenia, again, a side effect that was noticed in the previous randomized trials, as well as increased hemoglobin. As Paul mentioned, this was a drug initially targeting anemia, so it was an expected side effect, an increased hemoglobin. If we looked at the results, the serious events of increased hemoglobin was 0. Now, it's true that 13.8% had an increase of hemoglobin that was more than 2 grams per deciliter above the upper limit of normal. But the severity of the side effects due to increase in hemoglobin were mitigated by the protocol where the dose could be halted or decreased due to an increase in hemoglobin. So I think the strategy of following hemoglobin as well as adjusting or holding the dose is an effective one. If we look at thrombocytopenia, there was 6.1% of patients developed any level of thrombocytopenia, but really the serious thrombocytopenia occurred in 3 patients. None of them required stopping the treatment, so they continued on sotatercept with resolution. And then lastly, telangiectasia, an effect that was noticed initially in the phase 2 open label extension trial. So it suggested a longer exposure to the drug rather than an acute exposure to the drug occurred in 17% of patients. The majority of these events were evaluated by the investigator as being related to the treatment, so it was no surprise. However, there are no serious events of telangiectasia, and one patient only discontinued the treatment due to this side effect. When we try to look at any correlations or associations with background therapy, we couldn't find any signal regarding the concomitant use of anticoagulants, prostacyclins, whether there were any prostacyclins or parenteral or any other therapies. We tried to understand whether longer time exposure to sotatercept leads to increased risk of telangiectasia. This is a Kaplan-Meier curve that suggests by flattening the graphs in time that there is a lack of accumulation in the risk of side effect. However, we have to recognize that as the time increases, there were less patients that were exposed longer term, and this is only an interim analysis, so further analysis down the road will give us probably more accurate information. As far as summary of clinical worsening events, there were 20 patients who incurred 22 clinical worsening events. This is the table. None of them were deemed related to the study drug, but rather to the advancing of the disease. A few words about clinical efficacy measures. In the first part of each graph, it shows patients who crossed over from the placebo arm in the parent study onto the active treatment in Soteria. The second portion of each graph shows patients who had been on treatment in the randomized trial and have been continuing in Soteria. And then the first block shows baseline Soteria versus 24 weeks versus 1 year. And the first one shows the improvement in 6-minute walk distance in the former placebo patients once they started in Soteria, with maintenance at 1 year, as well as maintenance of the improvement in the walk test in patients continuing sotatercept. The same trend showed in NT-proBNP levels with improvement in placebo cross and maintenance in continued sotatercept. The first graph shows improvement in functional class, the proportion of patients achieving functional class 1 in green and 2 in yellow, increasing in the placebo-crossed and continuing in the sotatercept. And then the last graph shows the proportion of patients that achieved the low French risk score in the green portion of the bars. The more patients achieved a low risk score once they switched to sotatercept, and they maintained this achievement over 1 year. If we look at the changes in background prostacyclin therapy, we noticed that 17%, so 4% of patients required an increase in prostacyclin of at least 10% of the dose. However, we also noticed that 15% discontinued parenteral prostacyclin, and that it's an addition of patients who discontinued and transitioned to and de-escalated their prostacyclin treatment. So in conclusion, long-term treatment of sotatercept in Soteria was generally well tolerated, with less than 5% of patients having adverse events leading to treatment discontinuation and death. The 3 important side effects were telangiectasia events, thrombocytopenia, and bleeding events being the most common. However, less than 1% led to treatment discontinuation. There were improvements in clinical efficacy at 1 year, and with a caveat that this is the first interim analysis and more patients will be enrolled and we will learn more in the near future. I would thank you for the presentation.
Thank you, Anna. Again, elegant talk. So the last speaker for this morning will be Olivier Sitbon. He will be talking about seralutinib in PAH, treatment, mechanism, and long-term observation outcome. Olivier.
Thank you very much, Abdullah. So good morning, everyone. Now we will discuss the seralutinib in PH therapy, and that's my disclosures. So you have already seen that picture shown by Mark on the mechanism of and the implication of PDGF and its receptors in the pulmonary arterial hypertension. We know that there is an aberrant PDGF signaling in PAH and an increased expression of PDGF and receptors in patients with PAH, and this drives the pulmonary vascular remodeling. So there is a potential to reverse remodeling therapy by inhibiting this pathway, and this has been shown in experimental model where imatinib reduced the PDGF-induced small muscle cell proliferation, as you can see on this slide. So the first attempt to target the PDGF pathway was with sorafenib, which is a multikinase targeting both PDGF and VEGF receptors, tyrosine kinase signaling, and in experimental models, it works. It works very well in animals, but when we try to use that drug in patients with pulmonary hypertension, it is a small trial in 12 patients. We observed a non-significant improvement in 6-minute walk distance, but a change in cardiac output, and the cardiac output decreased with sorafenib. At the end, there was no change in pulmonary vascular resistance. Second attempt was, of course, imatinib. Imatinib as a treatment option for PAH, and there were a phase 2 trial showing an improvement in hemodynamics with imatinib, a drop in pulmonary vascular resistance. And after that, the IMPRESS study was launched. It was a phase 3 trial, and in the phase 3 trial, IMPRESS, we observed with oral imatinib an improvement in exercise capacity and also in hemodynamics in patients with advanced PAH, for patients having high level of PVR of more than 10 Wood units. However, we observed serious adverse events with the drugs, and the rate of discontinuation was very, very elevated. 33% of patients discontinued with imatinib, and this was mainly due to the occurrence of adverse events. And finally, the development of the drug in PAH was not continued. That's why some other formulation of imatinib have been developed, in particular via inhaled route, and much lower dose of imatinib have been tested as compared versus placebo in the IMPACT trial. Unfortunately, the IMPACT trial did not meet the primary endpoint of change in pulmonary vascular resistance. So if I stop here, you can say, okay, this pathway is not the right one to target. However, we have another drug, which is seralutinib, which is a multikinase inhibitor acting not only on PDGF receptors, but also in CSF1 receptor, c-KIT, and also having interaction with BMPR2. So the drug is able to reduce inflammation and vascular remodeling, proliferation, and also fibrosis. The first trial was the TORREY trial, which is a phase 2 study. It was a randomized, double-blind, placebo-controlled trial, and 80 patients have been enrolled in that trial. It was a 24-week trial, and 40 patients received seralutinib twice a day by inhalation, as compared to 40 patients receiving placebo. This is the baseline characteristic. It is a very, very busy slide. This is very, very consistent what we observe in other trials. The mean age was around 50, vast majority of female, majority of patients with idiopathic or heritable pulmonary arterial hypertension. But as you can see, we had an imbalance in the functional class between placebo and seralutinib. And as you can see here, we have much more patients in functional class 2 in the seralutinib group as compared to the placebo group. In terms of background therapy, the majority of patients receive a heavy treatment with triple combination therapy in 57% of patients, and 44 of them receive parenteral prostacyclin. This is the primary endpoint, the change in PVR from baseline to week 24. As you can see, we observe an improvement in PVR in the group receiving seralutinib as compared to a mild deterioration in patients receiving the placebo. The change was, the treatment effect was 14.3%, that is, was significant. But interestingly, the effect was more pronounced in more severe patients, functional class 3. We observed almost 21% reduction in pulmonary vascular resistance. And when we look at the effect in the pre-specified subgroups, this was very, very consistent according to the sex, to the number of background therapy, of prostacyclin at baseline or not, the age groups, the region, PVR strata, functional class, etc. So we have a very, very consistent effect according to these different subgroups. Let's move now to the secondary endpoint. Secondary endpoint was the change in 6-minute walk distance. Of course, the study was not powered to demonstrate an improvement in 6-minute walk distance. And what we observed, it was not significant improvement in 6-minute walk distance in the overall population receiving either placebo or seralutinib. However, when we look at the most severe population, patient functional class 3 or patient having a high reveal risk score, those patients had a much better improvement in the 6-minute walk distance, and this was plus 37 meters, and this was significant. Again, this analysis was pre-specified. Look at the NT-proBNP, which is also a very important marker of RV dysfunction. What we observed is a progressive increase in NT-proBNP in patients receiving the placebo as compared to a decrease in NT-proBNP in patients receiving seralutinib, and this was obvious as early as the week 12, and this was continued until the week 24 with a difference of 400 nanogram per liter. Change in other hemodynamic parameters, we have the change in mean pulmonary arterial pressure. We observe a significant difference between the seralutinib group and the placebo group. And of course, this was also seen in stroke volume, in cardiac output, and in PA compliance, which is a calculation between pressure and flow. Adverse events, the drug was well tolerated, and the only side effect, main side effect observed was the cough, but as you can see, we observed about the same proportion of patients who had cough on placebo or in patient on seralutinib. Otherwise, it was very, very well tolerated, with some patients having diarrhea, dizziness, but nothing very, very significant. Let's move now to the open-label extension. We had 3 objectives of the open-label extension studies. First, the long-term safety and tolerability. It is the main objective of the all open-label extension trials. Number 2, the characterization of the efficacy profile of the drug through detailed analysis of patients with PVR improvement from TORREY baseline to week 72. of the open label extension, and finally, to characterize some circulating biomarkers responsive to seralutinib therapy. Let's start with the study designs. The 74 patients enrolled in the there were 74 patients enrolled in the open label extension. 73 will complete the Torre +1 from the phase 1b study, and the hemodynamic data were available for 55 patients at week 72. And we analyzed, we focused our analysis on patients treated with seralutinib for at least 72 weeks, 28 patients, and we also defined the patients who had a more than 15% reduction in PVR as responders. Of course, those results are purely descriptive. This is the baseline characteristic, nothing very important on that, just to show you that, of course, the patient receiving seralutinib in the double-blind phase of the study, they were less severe at the time of enrollment in the open-label extension. It is something that we expected. Regarding the tolerability, safety and tolerability, there were no new safety signal associated with seralutinib in the open-label extension, and the treatment was well tolerated. Only just 3 patients had an increase in liver enzyme, and this was reversible. And in all 3 cases, we had another explanation for this increase in transaminase. Hemodynamic improvement. Some of those patients, 27 patients, they have been recatheterized after 72 weeks. And as you can see, the PVR continues to improve with seralutinib in the open-label extension. And this was obvious in patients receiving seralutinib from the beginning, but also in patients who were placebo-crossed, even if those patients, the improvements observed was less pronounced in patients from the ex-placebo group. In terms of, this is the data of the patient receiving seralutinib from the double-blind phase. So we have, and those patients have been catheterized at week 24 and at week 72. And as you can see, that's individual data. We observed that the patients receiving seralutinib from the beginning, they have an additional improvement in hemodynamics on the long term. And the 3 red arrows showed the patients having very low pulmonary vascular resistance after 72 weeks, and those patients, they had less than 200 dynes, which is 200 dynes is 2.5 Wood unit. Of course, this was driven by both the change in MPAP, but also change in cardiac output. We have a nice improvement in cardiac output in those patients, and this leads to an improvement in pulmonary vascular resistance. 6-minute walk. 6-minute walk has the same evolution, and the 6-minute walk distance continue to improve after the week 24. You have on green, we have the patient receiving seralutinib from the beginning, and as you can see, the 6-minute walk distance continue to improve after 48 weeks and after 72 weeks, but also the patient receiving placebo, when they were crossover for seralutinib, they have an improvement in exercise capacity. Finally, we look at the biomarkers, and we look at the many biomarkers, and more than 300 biomarkers have been shown to have a difference between placebo and seralutinib, and just here I'm showing here 6 biomarkers that are associated with pathogenic vascular remodeling, inflammation, and fibrosis, but also hemodynamic improvement. And for example, FLT1 correlates with pulmonary vascular resistance, or angiopoietin 2 correlates with pulmonary vascular resistance, compliance, anti-proBNP, and also echo matrix. This is my last slide and conclusion. We had now promising results of seralutinib as a potential treatment of PAH. First of all, the phase 2 trial met the primary endpoint of reduction in PVR in a heavily treated patient study population on background therapy. The pre-specified subgroup analysis showed that a greater benefit in the most severe patient functional class 3, or having a REVEAL score of more than 6. The open-label extension data demonstrate a promising long-term efficacy profile up to 72 weeks, with a continued improvement in PVR and exercise capacity on the long term. The drug was well tolerated, with no safety signal emerging over the open-label extension treatment period. And you know that now the phase 3 trial, the name is PROSERA, is recruiting, and we hope we can complete the inclusion before the end of the year. So thank you very much for your attention.
Thank you, Olivier. Thank you for this new updated information. We can have now questions. We have 10 minutes for discussion, and please maximize the use of these experts in the field with the new information has been presented to you. We can start with Stephen. Go ahead, please.
Good morning. Well, first of all, congratulations to the panel. Fantastic presentations. I really enjoyed that session. It struck me that all of you had genetics in there, and obviously I have to come on and ask a question here. So Mark, you said, it's important to look at the genetics to identify those pathways. Paul, you nicely showed that some genes overlap with different conditions when we look at the TGF-beta signaling pathway. Iona, you then discussed the side effects that have been identified in the sotatercept trials in particular, which struck me because it's bleeding and platelet disorders, thrombocytopenia, and telangiectasia. So what do you do about the genetic background in these trial lists? Are you using genetics as a tool to better understand why these side effects happen? Is that a missed opportunity there? And from my point of view, the question, isn't that a missed opportunity for the entire community to already select and collect genetic data for more targeted trials?
Maybe
you?
I can try to start, and many people will comment further. So in fact, it's a very timely question, because in the phase 2 PULSAR trial, there was a predefined genetic analysis, and we presented the early data at ATS last year, and the paper is going to be published very soon. So in fact, it's not a missed opportunity, it was a planned analysis, but you know, genetic studies require time. You need to see the importance, the likelihood of the genetic defect which are discovered. But now, what I can tell you, and it's a little bit disappointing for you maybe, is that the sotatercept results are similar in BMPR2 mutation carriers and non-carriers. And I think there is a key message in that comment. It means that we don't target BMPR2. We target the activin pathway and the global TGF-beta system. So many people think BMPR2 might be more likely to respond to this type of drug. It's not the case.
Maybe I can reformulate my question a little bit. It's not particular. I mean, it's important to know the pathogenic mutations for pulmonary hypertension. But I think what we also need to understand is what's the other genetic background so that these side effects potentially occur. So there is genetics that drive thrombocytopenia, for example, or other bleeding and platelet disorders.
Yeah, it has been studied as well, safety and efficacy and NGS analysis. So at the end of the day, the associations are not clear. You know, with other drugs, drugs like SAPH2025, there were some pharmacogenetic down, but the data are not that supportive of anything strong. So I agree with you, we could do more. It's extremely tedious to do because you need a lot of ethics behind that. It's international, etc. But no, you have a good point.
Thank you, Stefan. Majdi, please.
Thank you all. This was a great session. Just a question. I know that this was raised during the ad board, but as pulmonologists, we always have an issue with inhaled therapy. We know that in asthma and COPD, those people, they have a lot of adherence issues. The patients, they don't use their inhaler frequently, and this is why we are moving towards single inhaler therapy now when it comes to asthma and COPD. Plus, in this part of the world where smoking is becoming more prevalent, so really airway diseases will be an issue there. If you have somebody with severe asthma or COPD and start using inhaled seralutinib, so would that be an issue to consider in those patients?
No, I'm also a pulmonologist. I know what is the difficulty sometimes to convince the patients to take every day, twice a day, sometimes 3 times a day, inhaled drugs. However, with COPD and asthma, that's not that severe disease as for PAH. I think we can easily convince our patients that it is crucial for them to take the inhalator. It's usually twice a day. Perhaps in the future we can have development to have a once a day inhalation, which will be probably much better. But honestly, the experience we had with inhalation drugs in patients with PAH, with very, very simple inhalators, I think it is doable. The problem is the more complex. I remember when we started with iloprost 20 years ago, it was a nightmare for the patient to take the treatment because it was very long. It was 20 minutes, 6 or 9 times a day. It was terrible, and in that case, the compliance was not good. But with very small inhalators, as it is developed now, I think it is doable. And it's also the case for the PH-ILD. Now the majority of the drugs that are tested are inhaled drugs with very, very simple inhalers. And I think that is the way forward.
Thank you, Olivier. I think the company is working now to make it 2 inhalations instead of 4 inhalations each time. So we'll see what happens. Athenaïs, please.
Good morning, everyone. I have a question for maybe Mark, because I know your point of view, Olivier. How do you imagine the future treatment algorithm? I mean, if the seralutinib confirms its efficacy and is finally marketed, do you think that patients will receive both sotatercept and seralutinib? Or do you think it's time for precision medicine?
Yeah, thank you very much. It's certainly time for precision medicine, but it's difficult to do it. So what we will do, I think, will be very brief. If we have positive data, it will be a very important add-on therapy, and it will have to be positioned together with what we currently have. And we have to generate more and more data showing synergies between currently available drugs. For example, sotatercept and seralutinib will be important. For precision medicine, right now, it's a bit tough to predict, but in asthma, we have done it, so it will be done in PH. So thank you for your question. I don't know if Paul wants to comment on precision medicine. It's good.
Dr. Ragda, please.
Thank you so much for the panel for this excellent presentation. So my question regarding sotatercept. So would you consider to do genetic testing for BMPR2 mutation before starting sotatercept? Or is this going to be wasting resources?
Yeah.
Thank you so much.
Very good question. Because our preliminary data and our early data from POSTER suggests that a mutation does not predict response. In other words, patients without a mutation responded in a similar fashion as those with mutation. And moreover, if we looked at the subpopulation of idiopathics, hereditary, and connective tissue disease, they all seem to respond in a similar fashion. And we know the connective tissue disease patients typically don't have a mutation. Then the recommendation is not to, my recommendation would be not to test for the mutation to make the decision whether to use sotatercept or not, but only for those idiopathics that require counseling and regular, the typical genetic testing. Yes, but Paul would like to comment on that.
I agree that BMPR2 might be low yield given what we know about the lack of differential efficacy. But I think going back to Stefan Graf's question about the missed opportunity, a lot of centers now have access to whole genome sequencing for unexplained syndromes. And I think what we really would like to know is if there's a difference in genetic background for those patients who are susceptible to some of these HHT-like effects. Do they have SNPs at HHT loci that may not give you the full-blown syndrome, but predispose to the effects of ligand inhibition? Or do they have variants at other loci that are not related to the pathway that could predispose? The cost of whole genome sequencing is about $200 to $300 now. I don't know how that would get covered by insurance, but some specialty hospitals do include this in their care for syndromic cases of disease, especially in pediatrics.
Yeah, but I would like to add is we also know that patients without an identifiable mutation in the BMPR2 gene have deficiency in the BMPR2 function. So there may be some underlying connections with other pathways that are abnormal that may predispose these patients to respond to sotatercept.
Thank you, Hanan, and then Dr. Ayman.
Thank you for the great session. I have requested sotatercept for 3 patients, and I'm concerned about the GI complications. Do we need to do any test to screen or as a precaution before starting this medication? Are these serious complications to be expected?
GI
complications.
She's concerned about GI complications, GI bleeding.
Oh,
any precautions, any pre-testing before you start?
I see. Yeah. Very, very good question. We have not found a subpopulation that is more predisposed. For example, one would think that maybe patients with connective tissue disease who already have abnormal systemic vasculature and AVMs in the GI tract may be predisposed. So far, based on our number of patients, there doesn't seem to be a signal in connective tissue disease. However, there is a tendency for more bleeding in patients who are on anticoagulants and or systemic prostacyclin. So more hits on the coagulation pathway seems to be putting patients at high risk. But the severe GI bleeds were actually the 4 patients were all idiopathics and none were connective tissue disease.
Dr. Ayman, please.
Thank you for the fascinating talks. My query is in the view of the true anti-proliferative approach, don't you think that we need to go more upfront, especially in some patients like idiopathic pulmonary hypertension with comorbidities, cardiopulmonary comorbidities, people with anemia? Is there any trials going through these specific groups, going upfront rather on add-on treatment?
Good question. I think it will be handled by Olivier.
As a fan of initial combination therapy, I think that probably we need more data on that, because today we don't have any data of early treatment or at the time of diagnosis to initiate this kind of drugs, sotatercept or seralutinib or perhaps others. But probably in the future, it is very, very appealing to consider this kind of approach. I would say in combination with the usual drug, the available drugs we have, in particular the oral drugs, endothelin receptor antagonists and PDE5 inhibitors. But it is very, very interesting approach, and I'm not sure that we can answer the question with the data of Hyperion, but you know that better than me. But because
No,
Hyperion, that's not really initial. That's initial, but not.
It's early. But your question is very timely. I mean, we have to do that.
Yeah.
We have to do that. We have no data, but it's a good question.
Very good. British now.
Thank you very much. And I know that I'm holding people back from coffee, but thank you for allowing me time. So, Paul and Mark, there's been a lot of interest in the lack of rise in cardiac output with sotatercept. Some of that's going to be related to hemoglobin. I mean, mixed venous saturations do rise. So, but I'm just wondering from your work on the BMPs, if you've looked at the heart and whether or not there's going to be some potential benefit from the anti-BMP9 in terms of having less effect, perhaps a negative effect on the heart. If that indeed is the case, is it going to potentially allow us to see both an improvement in pulmonary vasculature and also allow an increase in cardiac output, or does it even matter?
That's a great question. Thank you, Luke. So I do think it matters. I do agree that the hemoglobin increases probably change some of the Fick equation components in the denominator to offset the increase in oxygen delivery that you definitely do have. With the PVR dropping, the hemoglobin change might neutralize the changes in cardiac output through that denominator. But I don't think that's the whole story. We know from the SPECTRUS trial that there were improvements in RV mass that were quite robust. And my question for those investigators was, did they see a drop in LV mass? Now, that didn't seem to bear out. But what I can tell you from preclinical models is that sotatercept is a very good anti-cardiac hypertrophy drug. All the drugs in this family are actually very good anti-cardiac hypertrophy drugs due to their ability to inhibit the hypertrophic effects of GDF8, GDF11, and activin A. But I don't think that the drug can discriminate between good hypertrophy and bad hypertrophy. And what we saw in animal models is that sotatercept reduces LV mass as well as RV mass. And I suspect that that might be some of the reason for the decrease in cardiac output. We did PA banding studies, and we did thoracic aortic constriction studies. And what I just described might be helpful for HFpEF-PH or CPC-PH, because you are then unloading hypertrophied left ventricle. But for anti-BMP9, we do see reduction in RV hypertrophy in those models, only in pulmonary hypertension. With PA banding, with TA constriction, there doesn't seem to be a very powerful anti-hypertrophic effect. So my prediction then would be that anti-BMP9 would allow the cardiac output to increase without that direct anti-hypertrophic effect.
We'll hold you to that, and we'll remind you of that.
I think if we can get through safety, and if we can demonstrate efficacy, that this translates from rodents to people, then that would be my prediction.
Well, it was a very disorganized question, but a beautiful answer. Thank you very much.
No, it's great.
Thank you, Luke. Always good question from your side. We will extend the break. I really want you to interact with your colleagues for extra 15 minutes, because the last session we have plenty of time. I have a question for you, Ioana. Any more elaboration on the hemodynamics long-term, particularly cardiac output?
Yes, a very good question. The Soteria study did not mandate repeat right heart catheterization because the Stellar trial had a 24 weeks, and then the blinded portion extended even further until the end of the study to show, to measure a worsening. So we though have repetitive echocardiographic measurements that we will be looking at into more detail in the long term. So indirect non-invasive measures, we'll look into it.
We'll
yes.
see. Yeah, please.
Break a little bit. Listen, I just wanted to say I thought it was a fantastic session, and I thought really hopeful. And seeing, but the slide that stood out to me most of all was actually one from you, Mark, when you showed in 2003, Activin was sitting there. And it was sort of, what made me think was hidden in plain view. It was there, and yet we had 20 more years to go before we had brought that idea to hope for patients. And it just made me think sometimes that in science, it's more for comment actually, I suppose, from you all, is that sometimes things are hidden in plain view, and we end up by chasing the effects of the disease. We go down in metabolics, we go down in epigenetics, we go down all kinds of paths. But we have a disease that has a genetic base. And Paul's talk was absolutely fantastic. You can see so many opportunities that we might be able to delve in and twist and tweak and come up with a bespoke approach to treating patients. So just more of a comment, absolutely fantastic session is why I love this conference. But maybe a comment, Mark, about, you've seen, you and I have been in this game a long time, hidden in plain view, there are a lot of little targets that are out there, but sometimes we lose track of that and we move off into the wrong direction. But now I think we're right on track again. Comment.
Thank you very much, Sean. Always very nice comments, thanks. Clearly, sometimes it's too difficult to target something when you are not ready or when you have not completely understood. For example, in 2001, we were younger, and we had patients, families with the same mutations and with antagonist phenotypes. I remember very well when the New England Journal of Medicine paper by Richard Trambas was published. I was part of the group, and I remember traveling from villages to villages in France to collect sample from a single family. And for me, it was an eye-opener because I could see one patient with PH and the mother with HHT. Exactly opposite phenotype, carrying the exact same mutation. So for me, it remains a mystery, but with my neighbor here, we try to complete the story and to understand the fine-tuning, which explains why people with the same mutation, because a mutation is a simple message. You have the environment, you have the epigenetics again, and many other things interfering. But right now, and the irony that our patients treated with sotatercept, they look more and more like HHT over time. And some of them look like HPS. I mean, it's amazing.
Yeah. Can I make a comment? I don't know if we got sidetracked. I think Mark is right. I think it took that many years to have a deeper understanding of this very complex pathway. It just took so long since we discovered the gene mutation.
And
so
by the
thank
way,
you for the preclinical scientists, for the hard work.
Yeah. And by the way, when I was driving from village to village, I'm still in contact with the family.
Aww.
So it's good.
Good. I would like to have your last comment on, so we saw from Soteria a plateauing effect all the way to like 1 year later. But originally it was a good response anyhow. And we're seeing from the Serotonin study a continued effect, positive effect on PVR 6-minute walk test. I would like to have your comment on this.
Yeah, many drugs with strong effect show strong effect early. And it is the case in PH, it's also the case in asthma and in other conditions. For some reasons, there is a plateau over time in terms of function, maybe because we just target part of the story and in an advanced stage of disease development. So maybe it will be my next comment is that if we could intervene early and as early as possible, maybe we would obtain normalization. But in our current state of health care, we identify PH patients very late in the course of disease when they have more than 80, maybe 85% of their vessels occluded or regressing. So you're right, the plateau is a message. Maybe as the gentleman in the room and Olivier were discussing, maybe early intervention will show you better effect.
Great, outstanding session. Obviously, you really deserve 20 minutes break. So we'll be back at 11:05. So we are late by 20 minutes for this beautiful discussion, but we have a last session, so we have plenty of time. Thank you so much. We'll see you in 20 minutes.
Hello, everybody. I think we're going to start this session. Actually, it's my pleasure to co-chair this session with my colleague, Dr. Takesho Ogo from Japan. And because I'm doing karate, so he is preferred for me. So, konnichiwa. Before I start the session, I would like to declare that there is a parallel now 6-minute walk test workshop. So anybody interested, he can go now and join them. Again, it's my deep pleasure and happiness to introduce Dr. Eileen Lang. Dr. Eileen Lang is going to deliver a talk about surgical, medical, and multimodalities treatment, PPA treatment updates. The floor is yours.
Thank you. I hope people come back in. I'm really delighted to be here again. Thank you for the invitation. And I'm also delighted to be allowed to talk about the most successful treatment of pulmonary vascular disease in chronic thromboembolic pulmonary hypertension. This is my hospital, and I like to show it. It's an example of a big tertiary care center, and this is where SAPH should be treated. And I think it's one of the building blocks of the new guidelines to emphasize the team approach and the centralization of care for rare disease. This is my conflicts. Much of the information that I will be giving you is based on the International CTEPH Association driven registries. And you can see on the slide the first registry, which also already goes back to 2007, which was shortly after the foundation of this international group of people. The second is from 2015 onwards, and the third is actually ongoing as we speak, the team registry. And in between came the BPA registry, which was a worldwide 18-center spanning registry run between March 18 and March 22. So all this is a very useful resource, and we heard from the geneticists that collecting data and collaborating is really the basis for insights. My first question to ask in this area is, what are the treatment targets? And I have some interesting considerations for you. You can see this is a pulmonary endarterectomy specimen, and you know that major vessels are the real site of CTEPH pathogenesis, but then there's also segmental, subsegmental, small vessel disease. And there is bronchial arteries that are target for disease modification. You can see here a recent three-dimensional optical coherence tomography reconstruction of the bronchial collaterals as they are shown on the histology slide in the adventitial layer of the artery. And that is important for interventionists if they use or shouldn't use cutting balloons or very high pressures, because you could rupture those vessels. And at the bottom you see the networks of capillaries around the vessels that are filled with lesions, as you can see them here, and I've shown in the past the webs and bands that obstruct the artery. In the algorithm, it's very clear. We anticoagulate, and then the multidisciplinary team comes in. And first line, pulmonary endarterectomy shown here, and if that is not an option, medical treatment and balloon pulmonary angioplasty. And the team is really important. Here you can see this is an image of the hybrid room in Bad Nauheim, and the guideline recommendation is very clear. There has to be a multidisciplinary decision team working together. But how do they do this? And I think I have mentioned that in the last year, and everybody would agree, risk stratification is the basis of treatment decisions. And what is risk in CTPH? We go back to the guidelines, we find the risk score for pulmonary arterial hypertension, and people have used it for CTPH risk stratification. And now with the data available from the second CTPH registry, you can see that the predictors of all-cause death for pulmonary endarterectomy were in fact to be on ECMO or to be hospitalized to prior right heart failure. So these right heart risk factors are probably the most prominent here. If you look at the BPA predictors of all-cause death, it's again functional class. Part of this ESC risk score for PAH. And if you look at those patients who didn't have any mechanical treatment, again, NYH class, or WHO class, and right atrial pressure, all those standard metrics for right ventricular function. So we are not doing badly with the ESC risk score. But there is other things in CTEPH that matter, and I would like to submit to you that the lesions matter. It's very clear for balloon pulmonary angioplasty approach, and Professor Ogo will confirm. You can see anything that's not ring-like or web is associated with a higher complication rate and with less success. For the surgeons, they say we can do distal as well as proximal because they are all experts. Andrea Damini, who is one of the authors of this work, is a world-leading surgeon, as is Madani, Fadel, and De Perot, and what their names are, but there are very few people who really know what to do. I would submit to you that distal disease is probably more difficult. And then there is more. There is associated medical conditions, Crohn's disease, VA shunts, splenectomy, that do confer worse prognosis and also affect survival. There is cancer recently from the Japanese AC registry shown to be associated with a higher incidence of all-cause death in a large group of patients, more than 1,000 patients with chronic thromboembolic pulmonary hypertension. Furthermore, you can assess risk in the cath lab. This is an exemplary case that I like to use to show the complexity of chronic thromboembolic pulmonary hypertension. If you do vasodilator testing, as we do, that can predict prognosis. And we've published that in the past. If you decrease the mean pressure by 10%, this illustrates there is still open vessels to work with, and the prognosis is better. Furthermore, the more complicated PVR partitioning that relates to histology. So if you have a very flat upstream resistance curve when you inflate the balloon for wedging, this is associated with more vascular disease and with worse prognosis. There is more. If you do a left heart cath and filling pressures are increased, that is a predictor of outcome. Please factor that in your treatment decisions. So we have pulmonary endarterectomy, we have balloon pulmonary angioplasty and medical treatments. Seems easy, but which for whom? And I believe it is important to decide on the initial treatment strategy. And that has been really changed by the availability of BPA. And you can see here the lesion, which is a complex web, has been opened with brisk backflow. 3 months later, everything is different. The vessel is dilated, functioning, flow is brisk, pressure is down. This is owed to our Japanese colleagues who carried on the procedure from first attempts in Europe to a very refined technique. And outcomes, as shown here, is to be able to lower mean pressure by 40% and PVR really by 60%. I think all the centers are able to, once they have passed the learning curve, they are able to achieve that goal. And that we have recently shown the BPA register, but it's still under consideration, so I cannot show you the actual data. But the 18 centers participating in the BPA registry, they achieved similar results. There is a price to pay, and this is in 9% of sessions, 30% of patients. There is mainly hemoptysis, and it's part of the learning curve, which is 200 cases to achieve a very low rate of complications. And the predictors is unfortunately female gender and very severe hemodynamics, which is also illustrated in this registry format in PH medications prior to BPA. So which first? PEA or BPA? Pulmonary endarterectomy is really the method of choice for very proximal and very big clot. There's no question. There's no balloon that's able to deal with a 3-centimeter clot. You know, I'm very convinced about this. Results of pulmonary endarterectomy are excellent. You can see that that's already old. And look at what was published just recently. 94% in a registry situation where many centers are contributing, not only San Diego, not only Paris. We achieve 94% 3-year survival. And BPA is very close. In this registry was 92, in the BPA registry, 94. So mechanical treatments are the way to go, compared with only medical. SAPH is the trial that is ongoing, and we will start in Austria very soon. We'll be comparing pulmonary endarterectomy with BPA in patients who are eligible for both. So it's the more distal disease that we will be addressing. Can we do it at once? Can we do BPA, PEA at once? Yes, we can. Christoph Wiedenroth has established a hybrid approach. He's still doing it, but it's a very rare procedure, even in his hands. This was a pulmonary endarterectomy where he did on the table in the hybrid room a BPA intervention. Furthermore, there is the sequential approach as shown here in patients from the Paris group with mixed anatomic lesions, and they were doing BPA first. Well, they first did medical, then BPA, then pulmonary endarterectomy. And you can actually get very successful with this approach. But it takes a team, it takes the recognition of which lesion type fits which treatment. What about medications? Let's look at those. We could treat everybody first, as was suggested in the past by Adi Gofrani, and I was fighting him. And I was saying to him, you know, this isn't great to have only 25% decrease in PVR. It is something, but I'm not happy with this. And you may conclude, well, PH medications, not bad, because you won't bleed, you won't have an excess site complication comparing medications with BPA. And 2 studies, as you know, looked at this. I'm going to run through because it's already previous data. Mr. BPA, done in Japan, randomized BPA versus Rio. And you do know that the change of mean pulmonary artery pressure was more substantial in the BPA group. No surprise. The RACE trial did a similar approach, looking at patients in a multidisciplinary team, if they were eligible for BPA versus Rio, and they found the same, more substantial decrease in PVR. So we already know this, and that is the stimulus for combination treatments. How do we combine? In the PH registry, 70% of patients were on drugs, and 60% were on drugs after BPA, which may change as we learn how to take away the drugs. And I think Nazareno, we will be taking away drugs from patients after BPA. And that will be perfect, will not be dangerous. But maybe we should also study systematically. What about before BPA? The recommendation is you should be considered, but the foremost study to address this is initial dual oral versus standard of care, initial oral prior to balloon. It's actually a little step further because it already assumes you need 1 drug. This is the IMPACT SAPH trial, which we will hear soon about. Maybe Marc can comment, or Olivier, on the state of the analysis. Should be coming up soon, right? What about before PEA? Before PEA, there is really even less data than before BPA, because there is no signs of improved survival if patients are pre-treated, and there is actually evidence that patients are delayed in their referral. This is shown by the San Diego group, who is, of course, a defender of the invasive strategy. The bridging study was another ICA-driven study, didn't succeed. It was actually designed to study whether Rio before PEA was could decrease PVR even further and all the endpoints associated with this. But it didn't happen because of Corona and other problems. So let me summarize. In 2025, we treat CTEPH following a multimodality approach. And it's very important to have a multidisciplinary team deciding the initial strategy. And you could add later another approach, but the initial treatment has to be PEA, BPA, or a combination, a hybrid, a sequential combination, depending on all the factors that are coming up, risk factors for outcomes, and that is definitely hemodynamic severity, including vasoreactivity testing and partitioning, patient age, patient expectations, comorbidities, and I would like to list their cancer that was recently shown very impressively, left heart disease, heart and lung disease. CTEPH disease type, I think, is extremely important for interventional people, lesion location, lesion characteristics, we will see in a moment, in the cases that will be shown following my presentation, and associated conditions, which is splenectomy, thrombophilia, cancer is also coming in here, I think, as an associated condition. And of course, the level of the team expertise, the level of the learning curve, and the availability of medical treatments. I would like to point out the ISHLT ICA meeting that is coming up this April in Boston. I've taken the liberty to change this respiratory to a true CTEPH logo. Thank you.
Dr. Irene, would you join us?
So next speaker is Dr. Faiz Al-Ahmadi. So he's going to talk about the incidence of insulin resistance and obesity post-PEA. So please.
So, Assalamu alaikum, good morning everyone. First of all, I would like to thank the organizing committee, Prophet Dal'an and Mr. Al-Uraini for the amazing SAPH meeting. I would like to thank Mr. Paula Pinto for the help in the slides. As well as I would like to thank myself, actually. As I am the chairman of the scientific committee. Having said that, most of the work has been done by Prophet Dal'an. Around 99.99% of the meeting was done by Dr. Dal'an. So thank you again. So we had this, Dr. Dal'an in the clinic, he had this idea about, he noticed an increase in the weight for the patients after pulmonary endarterectomy surgery. So he got the idea of studying the metabolic dysfunction and insulin resistance and the long-term impact on weight and metabolic syndrome in patients with CTEPH who underwent surgery. So first of all, I will talk about the objectives, then the background, the methodology of the studies, the results, and I will conclude with the future perspective. And these data, actually, it's from our hospital at King Faisal Specialist Hospital and Research Center in Riyadh. So I will present the baseline characteristics of patients with CTEPH from the Saudi PH registry. It's our registry in King Faisal. I will evaluate the fluctuation of the weight and the BMI, metabolic syndrome, and the insulin resistance post-pulmonary endarterectomy. And we will evaluate the long-term outcomes of CTEPH patients post-PA. So, as I mentioned, the rationale behind the study that we have noticed an increase in the weight for a CTEPH patient post-pulmonary endarterectomy, and this is kind of contradicting our expectations. We thought that patient post-PA will have more functional capacity and they will do more exercise and their weight will decrease. And we are trying for the first time, actually, to fill the gaps as we have lacking of data explaining actually the metabolic syndrome with the CTEPH patients. If you review the literature, we have the obesity actually and the metabolic syndrome and insulin resistance being studied actually in the CTEPH patients. And pulmonary endarterectomy, as you know, it's the gold standard for the proximal CTEPH, as mentioned by Prof. Iron Lang. However, the effect of PA on the metabolic syndrome post-endarterectomy and the insulin resistance, it was not inadequately studied. So, upon reviewing of the literature, I found that from the San Diego group, they categorized their patients into 4 groups according to the BMI. So BMI less than 22, the first group, the second group 22 to 30, and the third group 30 to 40. And the fourth group, the BMI more than 40. And actually, they conclude that the BMI doesn't significantly affect the outcomes after pulmonary endarterectomy. That study actually focused on the post-PA complications like the atrial fibrillation, the site wound infection, and the reperfusion injury. So no difference between the groups of BMI. Another study from UK, this is from the international UK cohort. So again, they categorized their patients into 4 groups. And they studied the main points after the endarterectomy were the hemodynamics, the 6-minute walking distance, and mortality at 5 years. Again, they didn't notice any difference in terms of the hemodynamic numbers among the BMI strata. Just important to mention, BMI more than 50 on that study were correlated with the bad functional capacity after endarterectomy and with a less survival on 5 years follow-up. So our study is a retrospective observational study using the Saudi PH registry. We included all CTEPH patients and we excluded all other types of pulmonary hypertension. We collected the demographics and the BMI, the WHO function class, pro-BNP, and hemodynamics. And we obtained the metabolic biomarkers, like we obtained the blood pressure, the diabetes, the C-peptide level, and insulin, and the lipid profile. And we chose the C-peptide and the insulin level just to refresh your endocrine information. The C-peptide, actually, it's very studied endpoints in the endocrine literature. So the high C-peptide level, the more that the chance you are having metabolic dysfunction and obesity. So it's an indicator of inflammation, the C-peptide. So we defined the baseline point. It's the point of the entry of the registry. So in the Saudi PH registry, we collected the data from December 2007 up to July 2021. And the last follow-up visit were 2023. And we have several and frequent visits of the patients during that period in our clinics. So you can see here the dashed red line is the time of pulmonary endarterectomy surgery, and that actually corresponds to time of 0. It's the time of pulmonary endarterectomy. Then the initial cohort, we have like 81% of the patients in our registry underwent pulmonary endarterectomy, and with a mean time of 243 days post from the baseline visit. Our mean time follow-up for the patients around 6 years, and the maximum follow-up that we have in our clinic around 15 years, and the lowest follow-up in the clinic was around 1.5 years. So at the end of data collection, none of the patients actually, all of the patients were alive, and none of them actually required neither hospitalization nor atrial septostomy. So this is quite complex. Just to simplify that, the green ones are the patients who underwent pulmonary endarterectomy. The red ones is non-PEA group. So the most common, this is the past disease profile. The most common past disease, of course, was the pulmonary embolism and the DVT. And around 42% of our patients have both conditions at a baseline. So how to read this graph? If you look to the first column, the green one, so we have like 9 individuals who had only PE diagnosis at the initial visit, and 8 of the 9 individuals underwent pulmonary endarterectomy, and only 1 patient, the one in red, he didn't underwent pulmonary endarterectomy. Again, if you look into details for the past diseases profile, we have DVT, antiphospholipid, patient with SLE, systemic hypertension, coronary artery disease, post-splenectomy, and etc. For the prescribed drug at the baseline, the most common drugs were the sildenafil and macitentan. However, around 35% at the baseline were not medicated with the vasodilator therapy. Then we have the most prescribed drugs at the last visit after pulmonary endarterectomy were macitentan and drasigwat. Having said that, again, 40% of the patients at the last visit were not medicated with vasodilator therapy. Most of them, they don't have basically residual pulmonary hypertension after endarterectomy. So we concluded 81 patients. You can see here, and 15 patients in the non-PA group and 66 patients in the PA group. Majority are female, and majority of them, like 35 patients in the PA group were obese, and at a baseline, and at the last visit, around 66% of the patients were obese after the pulmonary endarterectomy. Of course, most of the patients prior to surgery were in WHO function class 3. That statistically significantly improved after endarterectomy down to function class 1 and 2. Again, the median values of the BMI, I just want you to focus on the PA group here on your right side. So the median values of the BMI and 6-minute walking distance actually was significantly higher in comparison between the baseline values and the post-pulmonary endarterectomy values at the end of last visit. So the baseline was around 31, then it was 33 at the last visit, and the median value of 6-minute walking distance was 152 at the baseline, and it has improved up to 417 at the last visit. The median values of proBNP and thyroid stimulating hormone, it was significantly lower actually. comparing the baseline value in the PA group and the PA group at the baseline and at the last visit. So there was, of course, decline, and that's expected in the pro-BMB levels. And the thyroid stimulating hormone, it was decreased from 3 down to 1.9 post-endarterectomy. As I mentioned, we came with this idea years after the pulmonary endarterectomy, so no available data at a baseline for the C-peptide levels and the insulin. However, our study actually showed a trend toward decrease and reduction in the C-peptide levels in the PA group in comparison to non-PA group. That means you will have more of metabolic dysfunction and insulin resistance. Having said that, our study actually didn't show significant difference between the non-PA and PA group in terms of the C-peptide level and insulin level. However, data is ongoing, and we are collecting data. This is, again, box plot. It's explain what I just showed you in the tables. The red ones, as I mentioned, it's non-PA group, and the green one, it's the PA group. So the first column here is the difference in the BMI. You can see some of the patients here in non-PA group had dropped their BMI, and there was an increase in the BMI in the PA group after surgery. For the 6-minute walking distance, as I mentioned, it was expected that the 6 minutes improved after surgery. So this is the most interesting slide in my talk today. Here they used the linear mixed model. So it's a model that actually they have like 2 types of data, the fixed and random data, just to make it simple and short. So if you focus here in your right side, the dots actually represent the calculated BMI, and while the lines actually represent the expected BMI before and after the surgery. And the expected BMI, it was actually applied by the model. So overall, the data actually showed patients who underwent surgery with a high BMI, with the red one is BMI around 20, so patients who underwent surgery with a low BMI to start with, the model is expecting them to have a BMI higher after the surgery. As opposed to those who started with the high BMI to start with, around 45, the model actually expecting them to have a low BMI after surgery. And for instance, we have like calculated numbers actually. For patients who started with a BMI around 20, the model expected them to have a BMI around 24 at 5 years. And those who had a BMI around 45 expected to have at 5 years around a BMI of 43. So the limitations of the study, it's a retrospective design. It's the variety of the follow-up times. And as I mentioned, we have only unbalanced number between the non-PA and PA group. Only 15 patients on non-PA group. And actually, we used the BMI as the primary measures of the weighted changes. We didn't use the body composition, was not considered, like we didn't calculate the total fat, the free fat, the body water of the patients. That actually should be considered. So I would conclude with that. PA significantly improved hemodynamics and functional capacity, the pro-BMB and WHO function class. And BMI actually has increased significantly post-PA, and particularly in patients with lower BMI, and no significant changes in insulin or C-peptide level were observed post-PA. And despite cardiovascular improvements, metabolic syndrome components remained prevalent. And I would encourage all of you to have more studies on the metabolic syndrome in PH in general and CTIF in particular. So our future perspective, as I mentioned, the data is ongoing. We are collecting, again, the insulin and C-peptide levels for the new patients and the lipid profile. And future research that should consider the long-term metabolic outcomes, glucose intolerance, lipid profile. That actually could be a potential therapeutic strategies to optimize the metabolic health in CTIF patient post-PA. We can include more balanced subgroups and larger cohorts with more complex longitudinal designs. And by this, I conclude. Thank you.
Thank you, Dr. Faisal. Actually, very impressive work, and I'm sure it's going to stimulate a lot of discussion. And now, can I call Dr. Noor Al-Tourif? Dr. Noor Al-Tourif is going to deliver CTIF with antiphospholipid syndrome post-PA outcome.
We'll discuss.
Good morning, everyone. Thanks for having me. So today I'll talk about the post-PA outcomes in antiphospholipid syndrome. So I don't have any disclosures. So my objectives for today, I'll discuss key prospective post-operative complications associated with antiphospholipid syndrome after cardiovascular surgery. I'll review briefly the literature on APS-associated CTEPH post-PA outcomes, and I'll present the APS CTEPH post-PA outcomes observed at our hospital. So we all know that antiphospholipid syndrome is a complex and unique autoimmune disorder, and it's characterized by the persistent presence of antiphospholipid antibodies in addition to history of arterial or venous thromboembolism, plus minus a specific pregnancy-related complication. PE is the most prevalent complication and pulmonary manifestation in APS patients, and it accounts for 14%. And pulmonary hypertension has emerged as the second pulmonary complication in those patients. It accounts for 3.5% in primary antiphospholipid and about 1.8% in those with secondary antiphospholipid. And interestingly, around 20% of patients with CTEPH have positive antiphospholipid antibodies in high titers. So how would you diagnose antiphospholipid? So you need to have one of the 6 clinical domains plus a lab test. So you need to have either venous macrovascular complication like DVT or PE. You need to have arterial macrovascular complication like MI or stroke, microvascular complications like pulmonary hemorrhage, or obstetric complication, cardiac valve involvement, or hematological complication manifesting as thrombocytopenia, in addition to presence of antiphospholipid antibodies. So you need to have at least one documented clinical criteria from domain 1 to 6 plus positive antiphospholipid antibody in 2 tests 12 weeks apart. So we have a really limited literature on the post-operative complications in patients with antiphospholipid syndrome. And we have some data on the complications after cardiovascular surgery and mainly in valve surgery. And a lot of studies showed that there is significant morbidity and mortality in those kind of patients. Major complications were MI, acute heart failure, tamponade, neurological complications like seizures, CIA, or stroke, and the other complications that we're used to like coagulopathy, catastrophic APS, venous or pulmonary embolism. One of the studies in 1995 showed that 84% of their patients had a major post-operative complication, including thrombosed grafts, bleeding, thromboembolism, and strokes, and of course associated with high mortality rate. One of the studies published in 2022 by the Turkish group regarding the post-PA outcomes in patients with CTAF and antiphospholipid syndrome. And you can see that the majority of the patient had a functional class around 3, and they significantly improved after surgery to functional class 1. And regarding tricuspid regurg, the majority of the patient had moderate TR, and after surgery, they really recovered with trivial TR. And I think this is really reassuring that probably the surgery is beneficial in this group of patients. And these are the hemodynamics pre and post surgery in the same group. And you can see significant improvement in the mean PA pressure and PVR. However, they remained with some residual disease with a mean of 27 and a PVR around 3.4 to 4 Wood units. Another study was published in 2014 showed that they included 17 patients with APS and 190 patients controls. And they found that stroke and delirium were the most common complications after surgery in the APS group. Also, thrombocytopenia was really significant, and it happens from day 1 after surgery in APS group, and it was really significant at day 5 to 6 after surgery. The blue bar is the APS group, and the the blue bar is the control group, and the white bar is the APS group. So multiple studies and literature have linked autoimmunity in APS with distal vasculopathy and pulmonary hypertension. And therefore, we hypothesize that doing surgery in patients with APS CTAF they might end up with distal vasculopathy and more persistent pulmonary hypertension. And therefore, we did a retrospective analysis from 2011 till 2022. We included patients from the Saudi Pulmonary Hypertension Registry based on a single center experience at King Faisal. And we had 74 total number of patients post-PAA with CTAF, 37 patients with APS, and 41 without APS. We included adult patients 18 years of age or more with APS CTAF or non-APS CTAF who underwent PAA. The APS diagnosis was confirmed, was based on ACR criteria and was confirmed by a board-certified dermatologist. The CTAF diagnosis, of course, was based on the presence of chronic clots based on imaging and reviewed by a chest radiologist, and pH diagnosis was based on hemodynamic criteria from the 2022 guidelines. We excluded other pulmonary hypertension groups and those with other thrombophilia disorders like JAK2 mutation, protein C and S, and so on. Our first aim was to describe the baseline characteristics of patients with APS CTAF and non-APS CTAF. And we've included 37 APS CTAF patients and 41 non-APS CTAF. You can see that the majority of the mean age of our patient is really young, around 34 to 33, even when they don't have APS. And they had a lot of recurrent PEs and DVTs, but more pronounced in the APS CTAF as expected. The majority of our patients were females, as we always do. And the functional class was around 3 in both groups. And the exercise capacity was similar in both groups, but the ProBNP was worse in the APS group. Hemodynamics were similar in both groups, with a mean PA pressure around 50. PVR was around 12, with low cardiac output, cardiac index, and a similar mixed venous. And they really did have moderate to severe RV dysfunction prior to surgery in both groups. And around 50 to 60% of patients were on therapy before surgery, and 30% were on combination therapy. Our second aim was to determine the immediate and long-term post-PA outcomes of both groups, APS versus none. And you can see that most of our patients had the surgery done at our hospital. Years ago, we had some patients undergoing surgery in bigger centers at Europe and USA. post-operative hemodynamics showed similar numbers. Mean PA pressure was around 30, PVR was more than 2, and maintained cardiac output and cardiac index with similar mixed venous, and significant improvement in RV function. So regarding the characteristics of APS in those with APS, you can see that almost we have 86% with double or triple antibody positivity, suggesting that we have really highly thrombogenic group. And the majority is around 73% were primary antiphospholipid and 27 with secondary antiphospholipid. And the only connective tissue disease we had was SLE causing antiphospholipid. Hemodynamics after PA, as we would expect, both groups had significant improvement, and there was no difference between the two groups, except in mixed venous did improve significantly in the APS-CTEPH group compared to the non-APS. And regarding the immediate post-operative complications, we've decided to include hematological complication, mainly thrombocytopenia, metabolic complications like hyponatremia and acute kidney injury, neurological such as seizures, confusion, stroke, and cardiac like arrhythmia and pericardial effusion. We've defined the hematological and metabolic changes at day 5 after surgery to be related to surgery, and thrombocytopenia cutoff was less than 150, and severe thrombocytopenia was less than 50. Hyponatremia was considered if the sodium level was less than 135. So it's interesting to say that the APS-CTEPH group really had significant increased risk of immediate post-operative complications with a risk ratio of 1.56. Thrombocytopenia was common in both groups, but it was 90% in APS-CTEPH versus 45% in non-APS-CTEPH group. 23% of our APS-CTEPH had severe thrombocytopenia less than 50. 16% had AKI and hyponatremia, 16% had stroke, seizures, and confusion, and around 10% had cardiac complications. Regarding persistent pulmonary hypertension, so we all know that we don't really have a consistent definition for persistent disease. And we've decided to choose mean more than 30 and PVR more than 2 based on the first post-operative right heart cath within the first year after surgery, since mean of 30 aligns with our therapeutic threshold for those who undergo surgery and they continue to have pulmonary hypertension after surgery. So we have around 43% of patients with persistent disease based on our definition, without significant difference between APS and non-APS. And 32% of our patients in each group were on combination therapy after surgery at their last follow-up. Regarding mortality, we all know that the mortality in experienced centers is really low, and it's now less than 2.5, and it's really attributed to the advancement in managing cardiac and pulmonary complications after surgery. And prior studies have documented the safety of PA in patients with APS. And we have around 98.3% 5-year survival rate in our whole group. And there was no survival difference in CTEPH without APS, CTEPH with primary, and CTEPH with secondary APS, though we have really small numbers. Aim 3 was to identify predictive factors for poor outcomes, specifically in the APS-CTEPH group. We were interested in evaluating whether surgical timing was contributing to the outcomes, especially that we have different surgeons at different periods of our program, but it did not really affect the outcomes. The number of PE was of interest, so whether you have multiple PEs can cause you to have more persistent disease, and it did not really cause it. Pre-surgery hemodynamics, if you have more of a higher mean PA pressure or high PVR, also did not predict the development of persistent PH in either group. And interestingly, the APS-CTEPH group, when they got treated with pulmonary vasodilator therapy before surgery, they had lower risk of persistent disease with a risk ratio of 0.52 if they were treated and 2.65 if they were not treated. So in summary, 50% of APS-CTEPH group, they have increased risk of overall post-operative complications. Overall, 43% of our patients ended up with persistent pulmonary hypertension, regardless of the APS status. And the survival in our group is 98.3% at 5 years, regardless of the APS status. And this is our lovely pulmonary hypertension team. And thank you.
Thank you very much. PA seems safety and also effective even in the APS patient, it looks like. Yeah, thank you very much. So, next presentation is Dr. John Finchra from Australia, CTEPH in Australia and New Zealand. A deep look at the PH Sense registry. So please start.
Thank you. So I'm representing Australia and New Zealand today. These are kangaroos. They're actually Australian. With population-wise, they outnumber us. There's about 35 million of them bouncing around Australia. There's about 26 million Australians, 5 million Kiwis or New Zealanders. So I'm representing a number of different bodies, the Prince Charles Hospital, the Wesley Hospital where I work, as well as the Wesley Research Institute and the Pulmonary Hypertension Society. So when I was given this title for this talk, I rang my colleague Edmund Lau, who's the medical director of the PH registry, and said, can we quickly put this together as a deep look? There was silence at the end of the phone because we've just migrated over to PH Tool, and we're also doing a big data entry catch-up because of COVID and staffing across a lot of our centres over the last few years, plus this migration, where there's a backlog of data entry. So what I've done today is be talking about two of the major centres that we've got data from, particularly for the PEA. I'll be talking on behalf of Hayley Lewis, one of my colleagues, as well as ourselves at the Prince Charles Hospital. I'd also just like to say how welcoming it has been coming. This is my first time in Saudi Arabia with Professor Abdullah and Professor Hassan, and just how welcome that we've been and the things that we've been able to accomplish in a few days and visiting a number of the sites here. Definitely I'd be coming back to see a lot more. So these are my disclosures. So I'm representing the Prince Charles Hospital, so I'll give a bit of history with regards to the CTEPH in Australia. So prior to 2004, I was obviously a registrar and a fellow, and what we would be doing would be working patients up to go to San Diego to have their PEA if they required it. I think it was about $200,000 US at the time to get a patient across there. Then, rather than trying to save some money with that, is actually we flew Stuart Jameson out, so we'd line up, a week's worth or more of patients to have their PEA with Stuart Jameson coming and using our facility. So, and a number of those patients now, 20 years plus down the track, are still alive, running around, and they're known by their number, like they're number 6 from that cohort. So, and then from 2004, Professor Keith McNeil came back from Cambridge. So he brought John Dunning, a PEA and cardiovascular surgeon, back to Queensland, and we became the first operational CTEPH PEA unit in Australia, with that, bringing out John there. So Fiona was a consultant, a fellow, and then a consultant, and obviously they're the two that mentored me. So we're based in Brisbane, so that's in the southeast corner of Queensland. We're a population of just over 5 million for the whole state, and obviously for us, we're the CTEPH centre for the state, and we obviously have patients come from all over Queensland and also northern New South Wales. So Cairns is about 2,000 kilometres away from us. Until recently, we did not get referrals from Rockhampton upwards. Interestingly, I was thought that CTEF didn't exist in hot climates, but it was really about physician engagement and actually recognition. So when my colleagues moved into Townsville, did sabbatical, and all of a sudden we had this huge influx of patients coming down to see us with regards to CTEF management. So currently in Australia, we have 3 centres. Ourselves, Brisbane, Sydney and Melbourne. So at different times over the last 20 years, the main CTEF centre depended on the surgeons. So when John Dunning left to go back to Cambridge, we didn't have a surgeon. There was a surgeon in Sydney, so then all the PEAs went to Sydney. We've got Bruce Thompson, who was trained in Cambridge, and he's still with us. So we were able to, at our hospital, we were able to provide medical therapy, BPA, PEA, no surgeons in Sydney, whereas the Alfred, they can offer the same as us. Their program, I'll go through current management, I think we've sort of seen that today. Again, just the importance of the VQ. We still get referred patients that they say they've got PAH, but really they've got CTEF. We do the VQ scan and we go get into patients coming to us, the CTEF centre and our MDT. So within Australia, we do have two major MDTs. We do ours weekly because we sit down and have a look at what's been done the previous week with our interventional radiologists. The Alfred do a fortnightly one, and we have all of us discuss patients. Surgeons, as you know, may not always get to our MDTs, unfortunately, but we do discuss cases and give them first bite at the cherry if that patient's appropriate for PEA. So the 2 centres, Alfred, Prince Charles for pulmonary endarterectomy. So I'm presenting on behalf of Hayley Lewis now for the Alfred experience. They started their program in 2014 when Dave McGiffin came back from the University of Alabama to start for PEA and for their transplant program. And they became, because of that, sort of the main referral centre for other states, not just Victoria, New South Wales, and New Zealand, sending over patients for PEA. And you can see, even through COVID, the numbers didn't drop off. And the last data cut was, they did 31 in 2023. Sort of the usual demographics, I think, looking at female predominance here when we were looking at the CTEF, and I think that's the same in Japan, if I remember. It's not a 50-50 ratio, like we definitely are that when we look at our data. Usual comorbidities that we see with obesity, hypertension, and also with the antiphospholipid being down there at about the 27% of patients. Malignancy is also there. Typically prior to surgery, functional class 3, walk distance 415 metres, etc. And probably a lower use of pulmonary vasodilators going into surgery. Data from, again, pretty comparable for international data from the centre at the Alfred, with reductions in mean pulmonary artery pressure, PVR, residual pH being about 25% compared to the international data, 28 compared to 25%, and 30% of those patients with persistent pulmonary hypertension needing pulmonary vasodilators. Again, great survival data from the team at the Alfred. Australia, we've only got 30 million if you consider New Zealand. Sort of low volume compared to some of the bigger sites, like obviously Cambridge and San Diego, but good survival data. Our experience, obviously we started in 2004, we missed a surgeon for a period of time. Last data cut, it was about 150 cases. Since we started our BPA program, which I'll discuss a bit more, in 2017, we've done 25 PEAs. Over the life of the program, less than 4% mortality. And we've done BPA in 6 of our cases, and that's even 10 years post-PEA. And 30% of our PEA patients are on medical therapy. Again, this is old data, but we can again show that we've improved functional class, walk distance, pressures, etc., and quality of life. So we embarked on, when I moved to the Prince Charles Hospital, to look at developing our BPA program. I reached out to colleagues. Unfortunately, at that time, I didn't know Professor Matsubara or a lot of the people in Japan. So we reached out to our friends overseas. So our program is based on visiting 2 centres with Professor Nick Kim, with Mahmood. We also visited the Blue Group with Olivia Sitbon and Philip Bernot, and Nick Brown, our interventionist, also went and I think he did some balloon angioplasty on French citizens. So it was a very good experience for him. So last data cut at the end of last year, we had 184 CTEF referrals, went on to 175 diagnostic, as I said, 25 went on to PEA. At the moment, as of last week, we're up to 98 patients that we've had BPA and over 300 procedures. Patient age, 63, again, a bit of female predominance here, but BMI was 30. Main comorbidities is obviously the thrombophilia. We've got a number of patients that had splenectomy. Prior to the BPA program, we had a number of patients that were on triple therapy, including IV epoprostenol, because of their disease, and with ballooning we were able to get them off therapy. And functional class, predominantly 3 and 2 when they came through. We still have confabulating data with our registrar, because we've also had a number of interstate patients trying to get information from interstate, and also sometimes even within Queensland with the different medical districts trying to get information out of hospitals after someone's had the procedures. It can be quite difficult. The BMP's got the 2 asterisks there because in Australia until last year we had NT-Pro and BMP was not funded for pulmonary hypertension. We couldn't use it, or it came out of our cost centre, so that, and BMP. So there's not much change there, but obviously that's because we didn't have it there, but we can see that we improved walk distance, pressures, PVR, etc. Survival, exceptional, I think, when we look at it. The one death that we had was not related to the pulmonary hypertension or their CTEF after the procedure. Complication rate was down at 6%, mostly hemoptysis and vascular site injuries. So how do we compare? I compared us to when programs first started with the publications, obviously with the Japan, Germany and the French group when they first started, so we can see that we have a reduction with our BPA with 55% in improvements in pressure and 6-minute walk distance by 40 metres. So, I think my next slide is the new blue, the new hope. So our patients with BPA and medical therapy are getting to that stage of the Olivia Sitbon was talking about yesterday, that we've got these patients now that can be living for 10 years. So we can put out a risk assessment now, the blue Olivia Sitbon, very low risk, the hope, what we give these patients. Like Irene said, it is a team effort. It's not just me. Really highly dependent on our nursing staff for the coordination. We took on BPA without any additional funding. We've had excellent interventional radiologists and interventional cardiologists involved, our radiology people, our surgeon, and our colleagues in the thoracic department and cardiology. And just a picture of our team at our last ASM. I'm just going to promote, if you feel like coming out to Australia, we're a very welcoming country as well. Great food, great venue. It's in my hometown of Brisbane. You can see there Sean Gain enjoying himself there when he came out to us. And just that's at the end of the year, 23rd and 24th of October. Our main keynote speaker is Prof. Vakri. Thank you.
Thank you, Dr. Finestra. Actually, very distinguished experience. But you have to tell me, from where did you buy your suit? Now we reached the end of the session, so I think now we have the experts in the stage and experts in the floor, and it's time for questions and discussions.
Well, thank you very much. So great, great session, and thanks, John, for A, not wearing your suit, but B, also explaining the obscure joke from Olivier yesterday, which I think we were all waiting for the explanation today. So thank you. So it's a question for you, John, and maybe Irene, and anyone who else wants to pitch in, perhaps Olivier. I noticed that you have quite a high proportion of patients filtering through to BPA compared with PEA. You might say, well, it's not high, but in my experience in the UK, and I don't have the numbers, I think many more of our patients get filtered through to PEA. So I wonder if anybody has looked across different MDTs across the world and found whether there are different proportions of people going through to PEA versus BPA. I know in Japan, many patients go through to BPA, but that's potentially more of a sort of a cultural difference, potentially. So has anybody looked to see if there are fundamental differences in proportions of people going through to different modalities of treatment?
If I may respond, this is currently studied in the TEAM registry. That this is one of the main objectives of TEAM, to understand the MDT drivers for certain treatment pathways. I don't think currently we know, but as you have an excellent surgeon, the likelihood of having surgery in Papworth is higher. I think it's all depending on the team experience, but we will hear more about systematic influence on treatment decisions, I think. May also have to do with the population. I'm very surprised about the very young women with phospholipid syndrome. We don't see those. So I think it may be regionally very different and interesting to see from TEAM. I think that's really the purpose of TEAM. Please contribute.
Thanks.
John, Grant in Toronto. Thanks very much for an excellent series of sessions. Fayez, I was interested in your change in BMI. I mean, that goes very much against what our patients experience, who are volume overloaded, and then after surgery, their BNP and their heart function improves, and their water weight essentially decreases. In addition, I was surprised about insulin resistance, because most of these patients with heart failure have very high sympathetic tone as well, which would modulate insulin resistance. So I was a bit curious that there was an increase in BMI in your cohort of patients. And I'm just curious, you don't have any information about body composition, fat, muscle, water?
No, so this is, again, it's part of the limitation of the study. We didn't measure exactly the body composition components, like the total fat, free fat, the body water. So this is kind of one of the limitations of the study. We will consider that in the future. I think it's part of the increase of the body weight of our patients post endarterectomy. I would say the lifestyle, it's play a role in that, actually. So as I mentioned, we don't have all the baseline C-peptide level and the insulin level before the surgery, but we are ongoing of data collection for the new patients, especially for the C-peptide, the lipid profile, and the other parameters. Yes.
Right. Thank you. If I can ask one more question to Nora about her experience with the antiphospholipid antibody, particularly in your triple positive patients, those who develop neurological sequelae or severe thrombocytopenia. Did any of those get plasmapheresis either before the endarterectomy or afterwards? There's reports of using plasmapheresis or plasma exchange to manage severe antiphospholipid antibody.
Thanks for your question. So we usually evaluate our patients with antiphospholipid before they get admitted for the surgery. If they have any evidence of active disease, probably we'll do Plex and the other therapies to control the disease before surgery. But if they don't have any evidence of active disease, we just follow them up during surgery. And if they develop any manifestation, then we involve our colleagues from rheumatology and hematology. And for thrombocytopenia, usually we start the high-dose steroids and IVIG to manage thrombocytopenia.
in your cohort, how many of your patients then received plasma exchange or plasmapheresis?
Before surgery, it was the boss is here. He will answer the question.
Yes, there he is. Yeah, sure, you can answer that.
You will defend her, huh?
Yeah, Noura joined us 2 years ago. So yeah, we did several patients. We did actually plasmapheresis for, if I remember, 4 or 5 patients, or maybe a little less. And those, as Noura said, they had an active symptoms of active antiphospholipid syndrome before. Like one of them was actually thrombocytopenia. So we did it for a week, 5 sessions, between 5 sessions and 7 sessions, and then we got them into the surgery. But as you saw from the data, we have a significant number of those patients developing severe thrombocytopenia afterwards, and good number of them required IVIG plus steroids.
And use of aspirin, did you use aspirin at all?
After surgery, obviously, and before surgery, but perioperatively, of course, it was on hold. Yes.
So,
Dr. Ghadan.
Thank you for your valuable presentation. Question for Dr. Faiz, if you permit. What do you think about the dosage of leptin and leptin BMI? Are these parameters useful in your study? And data also.
I have to ask my endocrinology colleague. I don't have experience with the leptin.
I think this is
we have to ask them.
It's an important point, actually, when you discuss, because most of my patients lose weight after the operation, but I know you followed up for 6 years. It's a long time. So I think it's very interesting. Maybe we need to remove the bias of people getting more healthy, eating more, you know. So it's a very stimulating study, in fact.
So just one comment to add to the question of Dr. Granton. So maybe it's a good idea to have a genetic test for those patients, the CTEPH pre and post pulmonary endarterectomy. So in our hospital, we do genetic test for all patients for group 1 pulmonary hypertension. That's maybe if we can do more genetic test in CTEPH patients related to see if there is any correlation between the metabolic syndrome and the genetic predisposition.
Dr. Abdullah has something to add?
Yeah, I would like to thank Faiz in presenting data that is not really complete. And I think back to your note, John, we actually this BMI increase post-PEA seems to be a genuine finding. So what triggered us to look into this problem, that we really observed a significant increase in weight of patients 6 months to 1 year after the pulmonary endarterectomy, so which should not happen. It contradicts what we expected. These patients, when they have normal hemodynamics, they will have, you know, a normal lifestyle. Whatever they have, fluid retention in the past should have gone, and the weight should have come down. And that's why we, at that time, after our observation, we started doing metabolic testing, like C-peptide, insulin, and everything. But we've never done it for them pre-surgery. And this was like 2 years ago, and any patient we did from 2 years back till now, they actually have now a baseline C-peptide, lipid profile, and insulin, plus the other parameters. And we wanted to see down the road, a few years from now, to compare pre and post metabolic parameters. I have one small comment and a question to John. And if you see from the data Noura presented, we have a good number of patients actually having antiphospholipid syndrome. The last time we checked, more than 40% of our cohort are having antiphospholipid syndrome. And good number of those having very active antiphospholipid syndrome. So, and a few of them, in fact, had catastrophic antiphospholipid syndrome. So our cohort looked like quite different from yours.
Yes.
And I think you have like 8% of autoimmune disease patient, right? Which is very, very low compared to us, but more of hematological diseases, right? Yeah. Could this explain the fact that we have relatively higher post-surgery residual pulmonary hypertension than yours? You had more than 30, about 28%, and we have 40% post-surgery. Thank you.
Yeah, that could be. Yeah, I was thinking it's a different disease. Oh, I was just trying to think, yeah, definitely a different disease that you've got here compared to what we have in our cohort.
Dr. Lang, I actually have two situations I need you to give us your experience in. First is redo surgery. Suppose I have a patient who has a pulmonary endarterectomy, and after a few months he came with a pulmonary artery more than 40 millimeter mercury. Do you suggest to go to surgery again or to try PPA before we go to surgery, although he is taking the anticoagulants and the Risoguat and so on?
I think it's a common observation that you have, like 30% have post-PA pulmonary hypertension, in your case, pretty severe. So what I would recommend, and I think it's a general recommendation, is to work up the patient again. And we also work the patient up with invasive hemodynamics plus a PAG, pulmonary angiography. I think Dr. Ogo is also nodding. So we look for residual lesions versus other circumstances that may cause a more distal disease. You know, there is patients with significant microvascular disease. My personal take is the majority of those still have lesions. As great as your surgeon may be, he may forget some branches. So I'm highly motivated to offer BPA in those patients.
What's the time you allow before you do PPA in such patients?
That's a great question. I don't think we know. Usually the reassessment is done 6 months after the pulmonary endarterectomy. I don't think it's good to do it 1 month after, unless there's a very serious reason. And I would then not delay any further. We don't have any data that would tell us there's an advantage of waiting. There's usually an advantage of doing it earlier.
Dr. Agu, you have the same experience?
Yeah. I think we experience same thing. So we refer the patient to BPA after 3 or 6 months. Yeah, 3 or 6 months. But usually we waited until 6 months and to see if the hemodynamics improve or not. And then, yeah.
Another situation is acute and chronic. If I have a patient, and actually I had a patient like this, she was wrongly diagnosed as idiopathic, and she came with hemodynamic instability, hypoxia, and CTPA revealed that she has clear manifestations of chronic thromboembolic disease. We tried with her the anticoagulants, even thrombolytics, and the hemodynamics did not improve. Would you suggest surgery, and when, and would you go with PPA? It's a difficult situation, I know, but we need to have some experience in this. Maybe the floor also can help us.
Yeah. You know, I think acute PE is one of the best resources of finding CTEPH. So acute on chronic is CTEPH. Let's say CTEPH. So the acute on chronics is a term that I think shouldn't be used. It's based on the clinical experience that acute PE presents with a signature of chronic disease. And it's a fantastic resource to identify CTEPH cases. I think if you want to look for CTEPH in a population, go to those hospitals where they treat acute PE. And I think the acute PE mechanical treatments have been eye-openers for those interested in chronic thromboembolic pulmonary hypertension. So this is very positive, and I do think we all recognize that the PERT is also a CTEPH team. The pulmonary embolism response teams that are forming worldwide is another multidisciplinary team, of course, but I think it's the same. It's the CTEPH team with a few more people, with more the interventional cardiologists maybe, or the intensivists, the emergent care physicians, even hematology is participating now because they see acute PE, oncology. So it's a particular group that focuses on pulmonary vascular.
So would you, in this situation, would you give an optimum time before we go for surgery? I mean, trying the anticoagulants before the patient?
Yeah, that's a great question. I think the guidelines are reminding us, please anticoagulate for 3 months continuous prior to the decision. I think that is actually missing in the guidelines.
Even if hemodynamics are not improving?
Yes, you have to. You have to treat
patient
for
is
3
hypoxic,
months. But you may have already the taste of this could be an interventional case, so you can actually open the pathway in the meantime. You know, sometimes hospitals have delays, payments are delayed, you know, so you prepare. Don't treat with a vasodilator, but just use anticoagulation.
Yeah, I have another question.
Yes, Ali Abu Saud. I was doing BPAs at McGill, and just a small comment on one, the residual pulmonary hypertension after endarterectomy, as Dr. Lang described. It's very common that you go and you see exactly where that fibrous tissue broke. So it's really at that subsegmental level, and you see that there are the small branches that are there. So I totally agree. There are always lesions that are left behind, and they do very well. A little bit more difficult than, you know, fresh BPA or like lesions, but you can have very good results. And the second thing about the cardiogenic shock or, you know, hemodynamically unstable patients, this is also very common, and we can either, if you open 1 or 2 segments, it is dramatic how you can really get them over that hill, and then you can decide, do you go for surgery now or do you complete the job with BPA? But if you have someone who can really go and intervene on these patients, definitely much better than thrombolytic, which will give them only the side effects and none of the benefit of removing any lesions.
Yeah, good comment. Yes, we have another question, please.
Yeah, so I had a request in the break from the boss, Dr. Abdallah, to always ask a question about genetics in each session. So here we go. We've recently, as you know, undertaken a genome-wide association study in SAPH in a great international collaboration, and Irene, you contributed samples and were part of the authorship. We've identified that SAPH is partially heritable, and that there is common but very distinct genetic signals there shared between SAPH and pulmonary embolism and deep vein thrombosis. These findings, I think, are quite interesting. I'm not a SAPH expert at all, but can you say something about how useful these findings are in terms of, you know, treatment and potentially diagnosing, prognosis, etc.?
Well, I think it was a great effort that we had to do this. I'm still interested in mining it further. Yeah. I think for me, it was very gratifying to see blood groups, because as you know, we published this many years ago. We observed that VWF and the blood group phenotypes that are, I think, connected genetically. Factor VIII and blood group proteins are connected. So I think it is a confirmation for basic scientists to understand CTEPH. I don't think it can immediately be taken to practice, but I think it is a stimulus to move on and continue, even though we think that CTEPH is inflammatory thrombosis that may be triggered by other factors that are non-genetic. I think it is a great model for that. But we could discuss further, I think. Of course. I think it's acquired, you know. I think CTEPH is a great example of acquired.
Yeah. Great, thank you.
I just have a small question to Prof. Irene. So I believe there is a recent study from NICM about the genetic test in CETA. And Prof. Matsubara actually, for the time being, he's doing a big trial on the genetic test on CETF. What's your protocol? Are you ordering genetic test for all CETF patients?
I'm not.
Okay.
But we collect the samples. We collect Buffy codes, serum, citrate, and some circulating cells as well. Yeah. Because that is, actually we also collect platelets recently. And we do platelet testing. I think that's another very interesting subject that came from the Adam TS13 observation in the Toshner paper.
Yeah. Thank you.
Yeah.
Can I make a comment? About genetics in CTEPH? We have done genetic testing for CTEPH patients as well. And there are some patients who are not indistinguishable for normal CTEPH with that they got RNF213 mutations. So, and they got chronic thromboembolism as well. And we found that the complication of BPA is a bit higher than the non-mutation. So sometimes those kind of syndrome is migrated in the CTEPH diagnosis. So we maybe not CTEPH itself, but those kind of border the PH patient is migrated to as CTEPH diagnosis. So maybe to me it's maybe helpful to check the genetic test, to do genetic testing for those CTEPH patients to see whether the diagnosis is right. And maybe in the future, we can see more tailored diagnosis for CTEPH patients.
May I continue? So if you have an RNF213 mutation, do you call the CTEPH now in your reports, or do you call it PPS?
Yeah, that's a very very complex things. If it's a homo RNF213, it's obviously looks like very typical PPS, but it's if it's not hetero RNA mutation, in that case it looks like a very CTEPH phenotype. So, but it still has some features of vascular toxicity of PPS. So sometimes we misunderstand it. May not be misunderstood, but we can't distinguish between CTEPH or PPS. So
but no PA, that's for sure.
No PA, yeah.
Well, we have a last question, Dr. Yahya.
Thank you so much. I was a bit surprised that APS didn't add much more risk to getting a post-PAA persistent pulmonary hypertension than non-APS group. And from this point, I would ask, are there any risk factors pre-PAA that can predict that these patients specifically would have post-PAA persistent pulmonary hypertension, so that I can have a low threshold to start them on medication or consider them for sequential PPA post-PAA?
So I think we have a retrospective study with very small number of patients, so we can't say 100% we don't see difference between APS CTEPH and non-APS CTEPH. However, from the data we collected, I don't think we have a specific factor that can predict the risk of persistent pulmonary hypertension after surgery.
I think now we reached the end of this session and the last session in the conference, and I would like to thank all the floor and my colleagues and the faculty. I believe Dr. Abdalan is going to deliver his farewell remarks. So, Professor Abdalan.
BPA, same thing. They gain weight.
Well, thank you so much. It's the end of 3 successful, fruitful days. Let me, at the end, ask, of course, before I ask you to come for a group photo, like the usual and custom of SAPH, let me thank, first of all, the audience who are sticking to the last minute of a very exhausting 3 days. It means there is an interest in the pulmonary hypertension science. And in fact, this year we have focused a little bit more than usual on the basic science, especially genetics. And we really wanted to trigger the interest of young researchers, young physicians to choose this path. I think this path is the way to go. And at the end, we normally thank all those who participated as speakers or chairs or an organization. Particular thanks to our international colleagues who came from all over the world. Your presence has enriched the scientific level of SAPH meetings, but also your presence in our country was really a privilege, having you and networking with you and having with you good times. Thanks also to the pharma industry who participated and supported SAPH activities to the greatest extent. So thank you, all of you. I don't want to name some companies and I forget some, but thank you so much for making SAPH a real good meeting. I would like also to thank the audiovisuals for great work. And above all, let me thank my second-hand man, where is he? Hassan Al-Araini. Thank you so much for your great organization, making all this high-level scientific meeting happening. So thank you again. We will continue organizing SAPH every year, I hope, inshallah. And next year will be announced to you the location, the time. Normally the time will be around the same time as this one, but the location will probably be changed. At the end, we will take a group photo. I would like people to come here. Maybe the speakers can come on the stage. Our attendees come here, and then have a safe trip, every one of you, and we'll see you next year.
Good afternoon, everyone. There is lunch downstairs.
أهلا وسهلا ومرحبا. ما شاء الله الناس مبتسمة مروقة مو متوترة يعني this is something good الحمد لله. يعني في الوقت هذا عادة يكون وقت طويل صح؟ الدقيقة بتصير بساعة على بال ما تنتظرون النتائج. راح تكون إن شاء الله من الساعة 3 إلى 3:30 نعلن إن شاء الله على نتائج الفائزين في المرحلة أو التصفيات الأولى. ودي أذكركم إذا تبغون الوقت يروح بسرعة وتعلمون شيء جديد، عندنا المدرب معتصم الجوهرجي راح يتكلم عن المصمم المعتمد والتطوير المستمر، كيف تصبح مصمم سيرتيفايد ديزاينر. راح تكون إن شاء الله في منطقة ورش العمل، تبدأ بتمام الساعة 2 يعني الآن. فإذا كان عندكم مساحة من الوقت وأردتوا أن تتعلموا بشكل أكبر عن التصميم، إنه كيف تكون مصمم معتمد وموثق سيرتيفايد ديزاينر، راح يكون إن شاء الله معنا المدرب معتصم الجوهرجي يقدم ورشة العمل في منطقة ورش العمل. وبالتوفيق لكم جميعا. السلام عليكم ورحمة الله وبركاته. يا هلا وسهلا ومرحبا فيكم جميعا. ما شاء الله مروقين، يعني ما في توتر ما في شيء ولا؟ هذه هي اللحظة الحاسمة، لحظة التعب، الجهد، الوقت، الأشياء اللي بذلتوها، النوم في المصلى، الرقدة على الكرسي، كل الوقت اللي بذلتوه خلال اليومين الماضية كان عمل مهول خرافي جميل. يا جماعة الجو يجنن في الرياض ومطر وجميل، لكن الفئة اللي موجودة وجالسة في الجمعة بالليل إلى السبت الصباح، هذه أي فئة من المجتمع؟ أنا لما نقول والله عندنا فئة من المجتمع تكسر حاجز، تنجز، توصلنا ل 2030 طمحنا لها، هذه المجموعة اللي موجودة اليوم هي جزء لا يتجزأ من هذه الفئة المهمة في المجتمع. أشكركم من القلب، وأنا متوتر معاكم فما راح يعني أعطيكم قصة حياتي. هذه اللحظة الحاسمة لإعلان العشر فرق المرشحين للفوز في ديزايناثون 2025. لحظة حاسمة جدا تخليني أفكر كثير في هواجيس وأشياء ما أفكر. والله الدكتورة اليوم تقول لي استعجل، لا عاد تدرون عندي الأسماء وعندي كل شيء، يعني أنا بصراحة يعني خليني أقول لكم قصة لطيفة يعني قبل لا جيت اليوم الصباح. خلاص، هي اللحظة الحاسمة، اللحظة الجميلة، اللحظة إن شاء الله اللي أنتم كنتوا فيها أيضا جزء من التاريخ. راح نعلن عن مسار ابتداء من مسار صمم بإحسان. الفائزين العشرة مسارات انفيجن، نيرونز، فريق نزل، فريق وصال، فراجت، تيم سبارك، مركب، فريق وجد. ألف مبروك. ننتقل بسرعة المسار اللي بعده، مسار التحول الحضري. ألف مبروك. نقلة بئر 7، AC Innovators، مكانز، ستارت بوكس، أربن جيمز، VYB تيمز، ترانسفورمرز، وسم، التقاء. ننتقل إلى الاستدامة. العشر مرشحين للفوز في الاستدامة هم جرين برنت، ديتور، فيفا بلاي، خلاص تيم، ايكو شيلد، وفاق، كاب سايكل، فليبت، سستينيفيترز، رج هانج. ألف مبروك جميعا. ألف مبروك، تستاهلون كل خير. المرشحين اللي فازوا واللي جالسين حتى ما فازوا، يكفي جيتكم، كانت بصراحة يعني صنعت هذا الحدث بشكل كبير جدا. يعني أنا متحمس صراحة. أنا اللي عجبني أن كثير من اللي ما فازوا ولا ترشحوا ما شاء الله فرحانين أيضا بهذا الحدث الجميل. خلينا ننتقل للمرحلة القادمة والمرحلة الحاسمة والمرحلة الأهم، هي مرحلة التحكيم. الآن ركزوا معايا، سنخلي القاعة. أتمنى أن كل شخص يأخذ جميع الأغراض وتنتقلون إلى أماكن التركيز الموجودة في الخارج كل في وقته. طبعا الجداول راح تكون أيضا موجودة في الخارج، المساعدين التقنيين راح يكونون موجودين معاكم يقودون كل الفرق المرشحة للفوز أن يدخلون لمسارات التحكيم. ما أنسى أذكركم أيضا العودة للحفل الختامي إن شاء الله في 7 PM. ألف مبروك وفالكم التوفيق في النهائي يا رب العالمين. السلام عليكم ورحمة الله وبركاته. للتذكير مشاركين الأعزاء، حاولوا تأخذون جميع أغراضكم. ستخلى القاعة استعدادا للحفل الختامي على الساعة 7 مساء بمشيئة الله. للتذكير حاولوا تأخذون جميع أغراضكم، اللابتوبات، الحقائب. القاعة ستعد إن شاء الله للحفل الختامي على تمام الساعة 7 مساء. بالنسبة للغرف، المسارات والتحكيم ستعرض في الشاشات الخارجية أيضا للتحقق من أوقات تحكيمكم. بالتوفيق لكم جميعا. فزتوا ولا؟ أعزائنا المشاركين للتنويه مجددا التحقق من جميع الأشياء اللي موجودة الخاصة فيكم من فوق الطاولات تحت الطاولات بجانب الكراسي. سيتم إغلاق القاعة قريبا استعدادا للحفل الختامي. شكرا لكم.